to the day 2 of International Conference on Alternative to Animal Experiments and 6th Annual Meeting Society for Alternatives to Animal Experiments in India. I, Dr. Abhinav Tiwari, Junior Resident in the Department of Pharmacology,
I, Dr. Amrita Pandey, Junior Resident, Department of Pharmacology, JNMC.
would like to invite our chairpersons for the next session. In our esteemed chairpersons, I would first like to request Dr. Pratiksha Alag ma'am to come on stage. It is with great pleasure that I introduce Dr. Pratiksha Alag ma'am, our chairperson for this session, a distinguished professional with expertise in the realm of biopharmaceuticals. Dr. Alag has been at the helm of National Facility for Biopharmaceuticals over a decade, where her leadership played a pivotal role in advancing the frontiers of biotechnology as the in charge of this prestigious institution. She pursued her MSc in biotechnology at G.N. Khalsa College, where her commitment to excellence and passion for biopharmaceuticals were already evident. Today, as we welcome Dr. Pratiksha Alag, we celebrate not only her impressive professional journey, but also her unwavering commitment to pushing the boundaries of biopharmaceutical and research. Now, I would like to introduce our second chairperson, Professor C.R. Patil, sir. Professor C.R. Patil, Chairman of Department of Pharmacology, R.C. Patel Institute of Pharmaceutical Education and Research, an eminent figure with significant contributions to the field of pharmaceuticals and education, making him a truly distinguished leader in his domain. Professor Patil's journey in academia and the industry has been nothing short of exemplary. Sir is a seasoned scholar and a visionary who devoted his career to advancing the frontiers of pharmaceutical research and education, and under his leadership, the institute has flourished, nurturing the talents of countless students and contributing to the growth and development. Everyone, please join me in extending a warm welcome to Professor C.R. Patil.
Now I'd like to cordially invite Professor Tariq Muhammad, sir, who will be joining us online to be the chairperson for this session. Sir is the HOD, Department of Pharmacology at Integral University, Lucknow. Sir is a researcher in the field of pharmacology, a motivational speaker, a career counselor, and also a staff trainer. Sir did his Bachelor of Pharmacy at Jamia Hamdard and Master of Pharmacy at Integral University, Lucknow, Uttar Pradesh, and Doctorate of Pharmacy at Integral University, Lucknow, Uttar Pradesh. Sir was Principal Investigator at EMR project in 2018. Please welcome Dr. Professor Tariq Muhammad, sir. Our next chairperson for this session is Dr. Anil Kumar, sir. Sir, presently working as the HOD Department of Pharmacology, Gorakhpur. To be the chairperson of this session, Sir did his MBBS and MD from JN Medical College, AMU in 2000. Earlier, Sir worked in Maulana Azad Medical College, Delhi, Dharan, and Nepal GMC. Please welcome Dr. Anil Kumar, sir. He will be joining us online. Our next chairperson for this session is Abhay Deshpande, sir. Abhay Deshpande, sir, is the Global Director of Innovation and Strategy. Dr. Deshpande is a visionary leader and a trailblazer in the field of business and technology. With a career spanning more than a decade, he has consistently demonstrated his commitment to innovation, excellence, and global collaboration. As the Global Director of JRF Foundation, Dr. Deshpande has spearheaded numerous initiatives that has redefined the landscape of the company. They are in the process of development of predictive toxicology as well as efficacy models using 3D tissues, spheroids, and organoids with some of the top research institutes in India, Western veterans, and experts contributing these developments. Sir will be joining us online for this session.
I would request Dr. Pratiksha Alag ma'am to invite the speakers for this session. Thank you.
A very warm good morning to one and all present for this International Conference on Alternatives to Animal Testing. followed with the 6th General Body Meeting of the Society for Alternatives to Animal Experiments. Myself, Dr. Pratiksha, Head National Facility for Biopharma, would take this privilege to thank the organizers of the society as well as the conference and people at the Aligarh Muslim University for giving us the opportunity to come together and discuss a very critical and the topic of the future on alternatives to animal experiments. So in this session, I would first like to welcome all the other chairpersons who will be joining us online for this meeting. Welcome you all for this session. I'd like to now introduce our speaker for the session, Dr. Bharat Rudresh. He's a senior researcher at GRF Global. He's an in-depth and skillful computational biologist with proficiency in molecular modeling and simulation of micro and macromolecules. He has profuse expertise in using NGS and semi-supervised AI-based platforms. He has published over 25 research papers in many reputed journals and has been a mentor and co-guide for many students for the last 10 years. I now request Dr. Bharat to please deliver his session on the topic of developmental and experimental validation of the 3D QSAR model of thyroid disruptions using integrated method of computational chemistry. On to you, Dr. Bharat. Thank you.
Thank you very much for a very quick and nice introduction. Am I audible?
Very much, sir.
Thank you. Can you see my presentation? I would like to thank organizers of SAAE for giving us an opportunity to share our thoughts on animal alternatives. And in this particular session, I'll be talking more on development and experimental validation of 3D QSR models, which we have developed here for the screening of thyroid receptors using integrated methods of computational chemistry. And initially, I will be giving a quick background on the requirement of this study and what were the aims we had set before we start the study, and what is the catalytic side of the TPO, and what are the approaches we have taken to model the molecule, that is TPO, and its quality assessment and validation. And to give a quick background, thyroid peroxidase is an enzyme which is very crucial in biosynthesis of thyroid, and it is having a crucial activity in iodizing the tyrosine residues in thyroglobulin, and ultimately it gives a thyroid biosynthesis in T3 and T4 form. And in this particular enzyme, there are three different residues, that is aspartate, histidine, and aspartate at 238, 239, 240th position. They have their own activity, and these are the key positions we are supposed to look into. And the aim of this study was to model the structure of TPO, because there are no crystal structures for human TPO available in any structure databases. So we have thought of modeling the structure of this TPO and build the QSAR model. And for building the QSAR model, we have used known molecules with a TPO inhibition activity in terms of IC50. And using this training data set, actually, we have gone for a 3D QSAR modeling. And we have also evaluated a model structure as well as the model quality, QSAR model quality, using experimental data. And this is what exactly the catalytic site I was talking about. And this is a peroxidase domain on our sequence human TPO. And here you can see the highlighted stick model. Here we have aspartate, histidine, and aspartate. These are crucial residues which are actually making a active site of a peroxidase, because aspartate always helps in flanking the histidine residue, and also provides it with the favorable electrostatic effect to make it as a better proton acceptor. So that's why this is a crucial active site. And while modeling, we have adopted a homology modeling strategy where we have looked for the homology sequences. Here we have used the myeloprotease from Staphylococcus aureus as a model template with the identity of 48.10 and with a coverage of 61%. And you can also look at the fifth template from the Boston, which was having a 48.86% and with a bit more coverage compared to the one which we have selected. The reason for selecting the myeloperoxidase is its coverage in a domain region was better. When we compare with the fifth entry, its coverage was out of the domain. That's why we have focused more on a first entry than the fifth one. And these are all the simple statistics which gives us information regarding the protein sequence which we have used. It was containing 576 residues. Out of them, we had proper distribution of charged as well as polar residues, and it was a huge molecule to model. So this is what exactly the TPO which we have modeled here. We have evaluated the model quality at two different stages: secondary structure as well as tertiary structure. The secondary structure of molecules here, you can see the helices as well as sheets, and their quality was assessed using a Ramachandran plot. In the Ramachandran plot, you can see the yellow, white as well as red region. Yellow and red region corresponds to the allowed and strongly allowed region, whereas white region corresponds to the non-allowed region. So here you can see 4 different quadrants. Each quadrant corresponds to particular secondary structure as per the pi and psi angle. In this, you can see most of our residues, the dots as well as triangle, whatever you can see, are the residues, and most of our residues are falling in an allowed region. A very few are falling in non-allowed white region. Majority of them were glycine with a triangle shape. Since the glycine is a very small amino acid, it has no tendency to participate either in a sheet or helix formation. That's why we can see them in a non-allowed region. So this kind of occupancy is accepted. So apart from this, we have seen more than 98% of the residues are falling in a allowed region. And to analyze in a tertiary structure, we have seen the RMS deviation of the residues. Here you can see the residue at the residue histidine at 353 position was seen with high RMSD, as well as phenylalanine in 485th position was also seen with high RMSD. This is actually a concern, but these were falling much far away from our active pocket. That's why we have considered this RMSD. But if we avoid having high RMSD residues, it could be a better model. And naturally, the molecules or residues will be in a L conformer in a protein. But here we have 2 residues, that is threonine at 487 and aspartate at 513 position, were in a deconformation. So again, these were much far from our active pocket, so we have considered it. And here you can see the MT simulation which we have carried out to evaluate the tertiary structure. So whether the folding is proper or not. So to understand the protein folding, we have from the MT simulation. In the MT simulation, we have solvated the model which we have developed in a water solvent. Here you can see the radical red and white patches. That is actually a water. So when we do the MT simulation, it was very stable. Particularly active pocket was very stable. Apart from that, we could see some deviation in a loop, but active pocket was very stable. So this has given us a clarity on the quality of a molecule. So we have considered the quality of a model which we have developed as good for further studies. So again, visual MD you have seen. So this MD was run for 120 nanoseconds, and here you can see the RMSD within 4. And the deviation was within 1 angstrom, that is between 3 to 4. Initially, it took around 5 nanoseconds to attain the equilibration. After that, the RMSD was very much stable throughout the simulation. And even the potential energy of a molecule was very well maintained. And further, we have selected our best TPO inhibitor from our data set, that was 1H124 triazole 3 amine as a potential inhibitor, and we have docked it to the active site, which is containing a aspartate, histidine, and aspartate, as I have mentioned, and we can see the proper docking. And we have used this conformation as a reference conformation, and we have docked rest of the molecules in our data sets. So in our data set, we had around 556 molecules with 468 active molecules and 88 inactive molecules in our data set. And the best IC50 was 1.75. The same molecule was docked, and the conformers were developed with reference to this particular molecule. And we have developed different QSR models, that is random forest, KNN, as well as field QSR models, and where we have used 139 training, molecules in training set and 51 molecules in test set. And this is what the result we have obtained in a KNN QSR model. The R square was 0.80. Though it is not such a great value when it comes to the diverse molecule data set containing a diverse molecule. Here we have not used the molecules of congeneric series. We have used a diverse set of molecules. That's why 0.80 is a acceptable value. So we have evaluated with 10 different molecules from the external data set, and we have done the prediction for them. And apart from 2-3 molecules, rest other molecules were giving out good predictions for KNN model. And we have also used the random forest model. And here you can see the better accuracy than the KNN model, because random forest model is a AI-based model. It fits well for our diverse data sets compared to the KNN. KNN actually suits for the congeneric series of molecule. Because of the data set nature, we are getting a better results than KNN in case of a random forest. And again, when in a field QSR is another QSR model. Here we use the field of a molecule rather than focusing on a structure of a molecule exclusively. Here also we could get the comparably good prediction. Excuse me. So here you can see the excuse me. You can see the correlation between in vitro and in silico data. Here in a field QSR and a random forest, the correlation was very decent. Whereas in case of KNN and SVM support vector machine, the correlation was not that great. And this is how the field QSR work. Instead of aligning the molecule, here molecules are not exactly aligned, but the fields are aligned. So this is how we have obtained. These are the 10 different external dataset molecules we have, and these are the values which we have predicted, and the red colored values are the experimental values. So this is how we have docked other molecules. External dataset molecules were also interacting with the TPO model TPO very well. So with this, we have concluded that the model which we have developed for the thyroid peroxidase was good in terms of quality, even in secondary structure as well as in tertiary structure. And the QSR models which we have modeled for the prediction of TPO inhibition were more than 75% accurate to screen the TPO inhibitors in future. And the docked conformations are energetically favorable and depicts the quality of our active pocket as well as catalytic pocket. Thank you. And I would like to acknowledge my team members and leaders for their significant contribution. Thank you. I'm open for the questions.
Thank you so much, Dr. Bharat Rudresh, for such an extensive understanding of how QSR can be used as a very predictive model for development and analysis of new lead molecules, which can get into prospectively get into clinical trials. I'd now like to open the session for some questions. Do we have any questions from even for the people online? Is there anybody who would like to ask a question? Okay, Thank you once again, Dr. Bharat. Now we move on to our next speaker, Dr. Mamta Mishra. Dr. Mamta Mishra is a senior research scientist at JRF Global and has more than 10 years of experience in stem cell biology and regenerative medicine. She is an ardent interest in research pursuits with neurosciences and translational medicine being her field of expertise. She has over 25 publications in numerous national and international journals. She has exceptional analytical skills, attentions to details, and a penchant of thinking outside the box. She has made a go-to resource person within her research team right now. Now I request Dr. Mamta Mishra to please deliver her session on the topic of development and characteristics of the reconstructed human epidermis HPE model using in-house isolated HPEK comparative identicals to other epidermal models. Over to you, Dr. Mamta.
Yes. Can you hear us?
Yes, you're audible.
Is our screen visible?
Not yet.
Now?
Yes, it's visible.
Very good morning to everyone, and we are happy to be a part of Society of Animal Alternative to Animal Experiment, as we are the people among the million people to move forward in a positive direction. And today I'll be presenting the what we have developed at JRF, which is entitled as Development of and Characterization of Reconstructed Human Epidermal Model using in-house isolated human primary keratinocytes, which is identical to the other epidermal model available, which will contain the little bit brief introduction and how we isolated the human primary epithelial keratinocytes and how we characterize them, and then how we proceeded for the reconstruction of RHC model and how we have done the characterization. With that, we'll conclude and we'll show how we are going to move forward for the future directions. As we know, Russell and Bruce, they have proposed the 3R principles in 1959, but it took a decade of time to go for the non-animal testing methods, and European Union banned the animal testing of cosmetic products in 2013. Not only Europe, across the globe, we have a global trend, and we are trying to reduce the animal use for research and drug testing. At Joy Research Foundation, we are fully committed to 5R principles, and we select the appropriate model and pick up the right one that fits the new approach methodology. And always, as a European, we follow the requirements of the regulatory agency, and we establish the safe starting preclinical in vitro human state model. And I'll be presenting the reconstructed human epidermis model, which is in vitro human state models. We can see here the anatomy of human skin, and the top layer which exposed to air is epidermis, and it consists of 4 layers starting from stratum basals to spinosome, granulosome, and corneum. Two important points to say here that epidermis consists of the primary important cells, which is keratinocytes, which consist of more than 95% of the cells, and from the basal layers it undergoes differentiation towards the upper layer and stratum corneum formed, which is completely iron-nucleated cells. We can see here how we isolate the primary keratinocytes. As I have shown that the keratinocytes contains 95% of the epidermal cells. So how we have done it, with the permission from the Institutional Ethics Committee and ICMR, we have collected the human neonatal foreskin samples nearby hospitals, and then we isolated the human primary epithelial keratinocytes using the feeder-free and serum-free medium, and we subculture them and we have frozen them for the long-term storage. And then we revive those frozen vials to check the viability, and we have made enough number of stock. With that stock, we also characterize it before moving towards the RHE reconstruction. We can see here how we isolated the keratinocytes, and this is to show the morphology from the first week up to the end of the second week. And I would mention this important point here that we have isolated these cells using the serum-free media. And Dr. Ankita is present here, who will be happy to hear that we have always cultured these primary keratinocytes without serum media. And the end of the second week, we have the confluent cells, and we subculture them, and we have frozen cells with proper labeling and proper characterizations. We can see here we have used the marker CK5, which is a cytokeratin 5, and it's a cell-specific marker for the keratinocytes. On the top panel, we can see the CK5 expression is not there, which is used as a negative control. And in the below panel, we can see CK5 expression is more than 90%, 95%, which indicates that almost all the cells are expressing the CK5. And this is the overlay of the nucleus, which is shown in the blue color. And here we can see the blue and green overlay. To represent another proliferative marker along with the keratinocyte specific marker, we have done the double staining, and we can see here in negative panel also no CK5 expression, no Ki67 expression, but in the keratinocytes we have almost again 95% of expression of the CK5, which is keratinocyte specific marker, and CK5 is the proliferative marker. And in this panel we can see the overlay of the nucleus in blue color, CK5 is in green color and red color. Ki67 is green color and CK5 is in red color. And then we move forward and we check it whether these parameters are remaining stable for during the passage. And we checked all these passages from passage 1 to 3, and the stability of the viability which remains same from passage 1 to 3 as shown in the blue color, and the expression of the CK5 also remain constant, which is shown in the yellow color, and the gray color represents the Ki67 expressions. So to further move ahead, using this keratinocyte, whether the cells contains any fibroblast, we have checked that these keratinocytes isolated by us is devoid of fibroblast, which is shown here as a positive control. We have taken the human normal fibroblast and we have checked that it expressed the fibroblastin, which is a specific marker for the fibroblast. And in the below panel, which we have human primary epithelial keratinocyte, which doesn't express the fibroblastin, which indicates that these cells isolated are devoid of any normal fibroblast. Then we have moved ahead and we have chosen a number of vials before going to start the keratinocyte use in the RHE model. So we have a properly labeled keratinocytes vial, which indicates here the passage 1 and the batch 2 skin, and it has been isolated from the neutral skin. And we can see the numbers of more than 100 vials we have in the liquid nitrogen before moving ahead for the RHE models. So how we have constructed the RHE model? This is a schematic. We have the primary keratinocyte cells. On top of that polycarbonate insert, we have seeded the cells, and we have gone for the airlifting. And after differentiation, we got the RHE model. It takes around 3 weeks. So before moving here, I would like to mention that before moving to the airlifting, after seeding the cells, we have checked the uniform seeding here. As we can see here, uniform seeding is indicated by the DAPI, which is the nucleus of the cells, and these are highly proliferative cells, as you can see by Ki67 markers, and they are properly keratinocytes, which express the 97% of the CK5. So we went ahead and we have prepared the RHE model, and how we characterize, we are going to show that by hematoxylin and urostenin method staining, which shows the multi-layer epidermis. Then we have done the immunohistochemical staining for the specific layer, for the specific expression of the marker protein. And then we have checked the barrier function by trans-epithelial electrical resistance measurement. To show it here that this is the human neonatal foreskin tissue, where we have done the hematoxylin and biogenic training, and from this we have isolated the primary keratinocytes. And in the right panel, this is the RHE model developed in GRF. So as we can see here, this is a scale of 50 micron, and this is epidermis clearly visible here, and this is dermis part of the tissue. And from here, epidermis, we have taken the cells and we have reconstructed it. We can see here, this is more than 50 micron, and it has been published by different research group also. It increases up to even 78 to 80 micron, and we got also 78 micron thickness. To further move ahead, we have seen that this is the uniformity, and we can see these epidermal layers formed uniformly. It's not in one place, as we can see by dense magnification, and in the higher magnification, we can see all these layers are clearly visible. This is our polycarbonate inserts. On top of that, we have shaded the keratinocytes, and by the gradual differentiation, these layers were differentiated. We can see here the stratum basal layer, spinosome layer, and the stratum granulosum, and we can see the conified layers, which is completely anucleated and is shaded of the nucleus. Then we moved ahead to measure the epidermal thickness as well as the conified layer thickness from the day 14. It's around 2 weeks to 21st days, and we have seen that it conified layer is almost doubled by the 3rd week. And then we moved out and measured the conified layer as well as the complete epidermis, and we found that this epidermis thickness increases from the 1st week up to the 3rd week. By 21 days, we have more than 80 micron thickness, which is correlated to the other reported data in the literature. And then further we moved ahead and we have checked the cell-specific protein expressed in our RHE model. And this is the schematic. There are so many markers expressed in different layers, and we have picked up from the basal layer cytokeratin 14, and then the cytokeratin 10, which is in granular layer, and we have top layer filactin we have chosen to show the conified layers. And before moving towards our RHE, we have checked the positive control. This is the section of the neuronal foreskin as I was showing. We can see the filactin expression in the conified layer. It's a topmost layer of the epidermis. Then we have CK10 expression in the stratum, spinosome and granulosome, and CK14 expression in the basal layer. This green color says and blue color represent all over the nucleus. So this is our RHE model developed at RHE, GRF, which clearly indicates that this is the basal layer where we have the proliferating cells, and it expresses the cytokeratin 14, which is a proliferative marker for the keratinocyte differentiation towards the epidermis. So after this, we have checked also cytokeratin 10, which expressed the stratum granulosum spinosome. And here we can see the filactin, which is in the top layer, expressed in the conified layers. Then we moved ahead and we have measured the barrier function analysis by the transepithelial electrical resistance measurement by using the ohm. We can see here we have 96 RHE model, which all are going above the 200 ohm. So this is internationally acceptable wherever the RHE model, it's all published reports says that the resistance measurement above ohm signifies the formation of the functional barrier. Then to show that this is the gene expression analysis of the Ki67, which goes down from the monolayer to the RHE day 23. And here we have the filactin expression goes off towards the day 23, which indicates that the conified layers are properly called. And here we have to show that GRF India developed the RHE model, which is equivalent to the supplier 1 from France and supplier 2 from USA, which shows that properly formation of the epidermis along with all the distinct layer at the epidermis. And to conclude here, this human primary epithelial keratinocytes we have isolated. It has isolated in the feeder tree and serum media in a reproducible way, and it maintains the high proliferative capacity. And after viability also we have, after freezing also we have checked the viability, which retain its viability percent more than 95%. And these primary keratinocytes which we isolated used for the RHE model to reconstruct, and we have found that 4 distinct layers of epidermis are maintained throughout the RHE development. And this RHE maintains the barrier property, as we have shown that the tier measurement, and it is in a reproducible manner. And this RHE model developed at JRF is comparable to the other RHE model developed by other suppliers. For our future goal, we are going to develop this RHE model, use this RHE model for the skin corrosion and irritation test, and we are going to establish also the full thickness model of the skins, and also we'll go for the iPSC derived skin cells, because this time we have the isolated keratinocytes from the neuronal foreskin, and we are also planning to do the differentiation of the iPSC, and we'll do the 3D skin model and this period. And not the least, but we'll move out the microphysiological system and organoleptics for the future. Thank you. I'll be happy to take any questions.
Thank you so much, Dr. Mamta. First of all, I'd like to congratulate you and your team for developing such a beautiful indigenous model of the skin, which is as comparable to the industry international standards. So a great job and good congratulations to you and your team.
Thank you.
We'd like to open the session to some questions. Anybody from the audience would like to ask any questions? If not, then I have a few questions for you.
Sure.
Okay, please. Yes,
Please
yes.
go ahead.
Please, sir.
So good presentation, Mamata. Good morning to you, and good morning to all in the JRF group. I enjoyed listening to your discovery and your contribution to the 3D skin model, which is bringing up. I listened to you earlier, and this is much improved now. I just want to tell you that as you are aware, once you have a model, it should get it validated. That's the most important step in the progression towards the higher level. So you will have to find, Abhay knows that this is to be done, and it should be subjected to validation at some point. I wish our Indian effort for the validation in India comes through soon, in which case your product can be taken to that center for validation. Otherwise, you will have to do it in either it's Japanese or ICOM or ECOM, or somewhere you'll have to get it validated. So suggestion number one. And then I'm really overwhelmed with your ambitious plan to go into 3D from a stem cell, microphysiological system, organ-on-chip kind of stuff, and wish you all the very best, and wish you success in your endeavor. Thank you.
Thank you so much, sir. We have started the other work, and coming to the regulatory approval, we are on the process of doing all those proficiency chemicals, and on the way of approval from the regulatory agency, Dr. Deshpande sir and JRF will contact you, and we have recently heard that India is going to give us some clue, then we can register for the approval of from the regulatory agency. Thank you so much. Thank you, sir.
Okay, Dr. Mamta, I have a few questions for you.
Sure.
I'd like to know if you've done any karyotyping of the primary keratinocytes as a quality control of maintaining the genome number of the cells. So, have you done any primary karyotyping of the cells?
Yes, we have done the karyotyping of the cells, and also we have taken the control from other company to say that this is how their cells are behaving and our cells are behaving. So we have compared.
Okay. My second question is, when you use primary cells for isolation and culturing, the stability in subsequent passages becomes one critical issue. How do you think you're going to tackle that in your primary cell culture system?
Yeah, it's a good question. Yes, for the passage number, we have gone up to passage number 11, but for those primary keratinocytes, is used for other purpose, not the RHE purpose, because reconstructed epidermis needs the highly proliferative cells, and we have checked also the other publications and other resources how they made, and we have stick to the passage number 3. That's what I have shown. Passage 1 and 2 or 3 we have used for the RHE model, and we have enough number of vials to make the RHE using up to passage 3. So we'll use other higher passage for different work, but we'll stick to passage 3 for the RHE model. Thank you so much.
That's because sometimes the tissue source becomes scarce and difficult to obtain. So every time it becomes a very redundant expert. So I think that probably is going to be taken care if you go on to the induced pluripotent stem cell model. You will get more consistency and reproducibility in your subsequent batches over there. The other thing I would like to know that you've done a lot of profiling on your biomarkers to identify and characterize the RHE model. I'd like to know if you've done any proteomic profiling or Western blot studies to validate the expression and the consistency of these biomarkers.
Yes, we are on the process. We have the license. We are going to do that also. As many as biomarkers, we prove that it will be good for easier validation. So we are going to do that.
Okay. So thank you once again, Dr. Mamta. It was indeed a delight to listen to your presentation, and we wish you all the very best for your future prospects. Yeah.
Thank you so much. Thank you everyone present in the SAAE meeting. Thank you so much.
So we move on to our next speaker, which is Ms. Sri Rupa Mitra. It's my pleasure to introduce a remarkable individual who's been making a very significant contribution in the field of research and development at JRF Global. She is a shining example of dedication, innovation, and commitment to the pursuit of knowledge. She's embarked the journey in the world of research with an unquenchable thirst for discovery and a strong educational foundation. Her journey is a testament to the fact that with dedication, passion, and hard work, one can achieve greatness in the world of research. So I'd now like to invite Sri Rupa Mitra to deliver the session on the topic of QSAR-based mutagenicity prediction of nitrosamine drugs, substance-related impurities. Over to you.
Just give us 1 minute.
Yeah, hello everyone, good morning. Is my presentation visible?
Yes, very much.
Okay, thank you. So, hi, today I'm going to talk about QSAR-based prediction of mutagenicity for potential nitrosamine impurities. Dr. Bharat has already talked about QSAR study previously. So these are, as he said, that these are models which are mathematically and statistically developed, and they are used for chemicals for the prediction of chemicals toxicity. So here we use some of the third-party softwares like OECD QSR Toolbox, Vega, Opera, and LeadScope for these defined toxicological endpoints. And we can also build models, 3D QSR models for the target-specific toxicity and efficacy prediction. So nitrosamine impurities, I think most of you here have already heard about, like they know what is meant by these nitrosamine impurities. However, I will just briefly explain what nitrosamine impurities are. So these are compounds. Nitrosamines are the compounds which have nitroso groups bonded to amine groups. And these have been commonly found as impurities in drugs, and drugs like angiotensin receptor blockers, ranitidine, metformin. and they have been identified as potential human carcinogens. So this has raised raised safety concerns in the pharmaceutical industries and has posed a drug shortage risk. So US FDA has made it clear that a risk evaluation must be done for such nitrosamine impurities, and that this should be extended to all drugs or pharmaceutical any pharmaceutical products as a safety measure. So, due to different factors which are related to certain risk of contamination, so drug manufacturers are finding it difficult for evaluation of individual nitrosamine impurities. So, these are the 7 potential nitrosamine impurities in the commonly found in APIs and drug products. NDMA, NDEA, NMPA, NDIPA, NIPEA, NDBA, and NMBA. So, our objective in the study is to predict the mutagenicity of these 7 potential nitrosamine impurities in APIs and drug products using a QSAR read-across approach. We have used 4 different models and have derived the consensus with the results. And our aim is also to understand the coverage of data on the nitrosamine class of molecules in the datasets used for the prediction of mutagenicity. So the first model that we have used is the mutagenicity amidst Caesar model. The first molecule we have evaluated is NDMA. So here we see that in the training data set of the model, the experimental data for NDMA is already available. So it is identified to be mutagenic. Here we see the red dot. This indicates that the molecule is toxic, that is, it is mutagenic. If it was non-toxic, it would have been green in color. So the model has predicted the software in the model in the software has predicted it to be identified it to be mutagenic. We see that 3 compounds below. These are the compounds which are very similar to the molecule to our target molecule in the training data set of the model. We also see that it is the structural alerts here mentioned. This indicates that these are the groups which are responsible for the mutagenicity of this molecule. And we also see that the predicted compound is into the applicability domain of the model. So the next molecule is NDEA. Again, experimental data is available for NDEA in the training data set of the model. So it is already identified to be mutagenic. For NMPA, we also see that experimental data is available in the training data set of the model, and it is identified to be mutagenic again. For NDIPA, there is no experimental data available, and hence the model has predicted NDIPA to be a mutagenic molecule, and it has identified based on the similar molecules. As we can see that the below 3 compounds have almost 90% similarity, which are already identified to be mutagenic experimentally. So the model has also identified NDIPA to be mutagenic, and we see that the reliability is very high. We see that it has 3 stars, which indicates it has the prediction is highly reliable. Moving on, the next molecule is NIPEA. Again, the model has predicted the NIPEA to be mutagenic with high reliability. NDBA, again, experimental data is available for NDBA, and it is identified to be mutagenic. For NMPA, we see that the model has predicted this molecule to be mutagenic. However, the reliability is moderate because the predicted compound could be out of the applicability domain. That means some of the information in the training data set might not be available enough to show that the molecule is mutagenic. So still we see that the reliability is moderate, and this is the summary of the results that we have obtained. We see that all the 7 molecules are predicted and identified experimentally to be mutagenic. with good reliability. The next model that we have used is ISS model. Here again, we see that experimental data is available for NDMA and it is already identified to be mutagenic. For NDEA, experimental data is again available and it is identified to be mutagenic. For NMPA also, it is identified to be mutagenic experimentally. For NDIPA, ISS model has again predicted it to be mutagenic with very good reliability. For NIPEA, we see that the model has again identified the molecule to be mutagenic with good reliability. For the NDPA, experimental data is available and it is identified to be mutagenic. And again, for NMBA, we see that ISS model has identified, has predicted the molecule to be mutagenic, but again with moderate reliability because the predicted compound could be out of the applicability domain of the model. So this is the summary of the predictions from the ISS model, and we see that the model has identified all the 7 molecules to be mutagenic, and however NMBA has been identified with moderate reliability. Still it is predicted to be mutagenic as per the model. So the third model that we have used is SAAE-I IRFMN model. So NDMA, for NDMA, experimental data is available and we see it is identified to be mutagenic. For the NDEA, again, experimental data is available for the molecule and it is identified to be mutagenic. For NMPA, experimental data is available and it is identified to be mutagenic again. And NDIPA, no experimental data available. So SAAE-I IRFMN model has also predicted NDIPA to be mutagenic with good reliability. The molecules in the which similar to the target molecule in the training data set is already almost have 90% similarity with, and they are also identified to be mutagenic. So the prediction says that NDIPA is mutagenic. For NMPA, we see that again experimental data is available and it is identified to be mutagenic. NDEA, it is experimentally identified to be mutagenic. For NDMA, experimental data is available in the training data set of the model and it is identified to be mutagenic. So this is the for NMBA, no experimental data is available. So the model has predicted the molecule to be mutagenic, and we see that the prediction is with moderate reliability. So this is the summary of ISS model prediction of the predictions from the ISS from the wait. We move back, I guess. I'm sorry. So this is so this is the summary of SAAE-I IRFMN model. We see that all the molecules are predicted to be mutagenic by this model. So the last model that we have used is KNN read-across model. Here we see that NDMA, no experimental data is available for NDMA. So it is identified, it is predicted to be mutagenic by the model. For NDA, experimental data is available and it is identified to be mutagenic. For NMPA, we see that the molecule is experimental data is available for the molecule and it is identified to be mutagenic. For NDIPA, no experimental data is available. So the KNN read-across model has identified the molecule, has predicted the molecule to be mutagenic with good reliability. For NIPEA, the model has predicted the molecule to be mutagenic. For NDBA, experimental data is available and we see that it is already identified to be mutagenic. Here we see that NMBA is predicted to be mutagenic with good reliability by KNN read-across model.
So read across model is actually used for identifying the molecule for making predictions for molecules for which not much amount of data is available. So here, so NMBA for this, but this model has identified NMBA to be predicted NMBA to be mutagenic with very good reliability. So again, we see the summary here that KNN read across model has predicted all the molecules to be mutagenic with very good reliability. So this is the consensus that we have generated by using the results of all the models. And we see that all the molecules are predicted to be mutagenic. So in conclusion, we described in silico methods here to assess the MS test mutagenicity potential is found in the drugs and APIs. So all of these molecules and we see that there is very good confidence. Already experimental data available in the data set, and for some visible using the available data, we can predict the such predictions that they can be or not. So thank you, everyone. I'll be happy to take questions now.
Thank you, Ms. Srirupa, for the very extensive presentation on QSR modeling for mutagenicity. Is there anyone from the audience who would like to have any questions? Anyone online who would like to ask any questions? Chairpersons? Okay, so Srirupa, I may have one or two questions to you. So here we are talking about nitrosamine impurities, which are basically present in very small proportion in lead molecules. When nitrosamine already itself is a mutagenic agent, what was the purpose of estimating the impurities as mutagenic agents?
So, actually, or not all molecules are identified to be mutagenic. Some molecules, some of these nitrosamines are actually identified. So, this actually helps the pharmaceutical industries to, if this initial screening is done, so it will help to understand whether all of them are actually mutagenic or not. So, now not all of them are mutagenic. Some of them are mutagenic actually. So, this is just an initial screening step.
Okay. So how do you, all these are predictive models. Have you done any validation of these models to see how accurate you are to the original data?
Yeah, I would like to take this question, Bharat here. And this study itself is a validation study. Here, all the nitrosamines studied here are non-mutagenic. So whether they are really showing the mutagenicity in our prediction or not. So this itself is a validation study. So in future, if there are any nitrosamines identified in future, they can be directly validated for mutagenicity or for other toxicological endpoint using the same model. So we have understood the model efficiency or reliability to predict the present and future nitrosamines for different toxicological endpoints.
Okay, so if I may understand correctly, what you've basically done is already enough validation done for these. You've just seen a QSAR model, whether it is trying to fit the kind of validation data which are coming from the in vitro study.
Yes, the tools what we are using are providing the models, whether they are incorporating the nitrosamine data or not. If there is no data, then whatever the molecule we will be screening for will be with no information, and it may come out with a irrelevant result. So before that, we have to run the validation study. So this is validation.
Okay, I got it. Yeah. The last question that I'd like to ask you is, have you applied this model to any other mutagenic drugs and seen whether you're able to do it for other class of compounds or similar related compounds like nitrosamines?
Yes, we have done. we have also done using the negative molecules.
Okay.
These are the mutagenicity positive molecules. We have also validated with negative molecules.
Negative models of nitrosamines or any other drug molecule?
No, other molecules.
Other molecules.
Non-mutagenic.
Okay. Thank you so much for
Thank you.
friends. We wish you all the best, Srirup, for your future work. So we
Thank
move
you.
on to our next speaker, which is Jayanish Prajapati. Ladies and gentlemen, esteemed colleagues, it is my pleasure to introduce to you Mr. Jayanish Prajapati, who is a dedicated research officer at Jay Research Foundation Global for the past 8 years. Mr. Prajapati's academic journey has led him to attain his MSc in zoology from Sardar Patel University, setting the foundation for his impactful career in the field of research. Over his tenure at Jay Research Foundation, Mr. Prajapati has been instrumental in advancing understanding of various facets of cell culture, conducting both qualitative and quantitative studies, delving into the intricate field of toxicology. His commitment to research has yielded valuable insights, contributing significantly to the collective knowledge. Today, we are fortunate to have Mr. Jayanish Prajapati share his expertise in a session focusing on the in vitro inhalation toxicity assay of lambda cyhalothrin 5% EC, utilizing the cutting-edge epi-airway technology. Without further delay, I now invite Mr. Prajapati to the stage to deliver his insight. Thank you.
So first of all, thank you very much for the great introduction. And today I feel honored to be here to part of the great discussion, the topic of the alternative of animal testing. So once the animal testing considered the gold standard in a research field. But now we are standing at that crossroad where we have the opportunity to develop the new method while respecting the life, shaping the frontier of the science. So without much delay, we start with the presentation. So a crop protection product may induce damage to respiratory tract. This damage can be assessed by the acute inhalation toxicity testings. So from the recent days, the OECD guidelines are used to assess this kind of the toxic hazard induced by the various substance. So we take a step to develop an in vitro model of the inhalation study. So we use epi-airway model AIR 100. So these are the epi-airway model. This exactly look like the tissue culture insert. At the bottom of the insert, the monolayer cells were cultured. That cell have the ciliated columnar, goblet, and basal. So these are the few materials we use in this research. So lambda cyhalothrin 5% is used as a test item, as we know the toxic property of this particular compound. The formaldehyde 10 gram per liter used as a positive control. DPBS used as a vehicle, and epi-airway tissue, 3 tissue per group in DRF and 5 tissue per group in a main study used as a test system. So we use 100 microliter as a dose volume, and 6 various dose we selected for the DRF study. The exposure period for the research is 24 hours. And these are the few endpoints we consider at the end of the study. So MTT consider endpoint for the DRF study, and for the main study we consider the MTT, TR measurement, LDH assay, and histopathological examination. So now we come to the part of the procedure. So upon the arrival of the tissue, tissue transferred to the pre-incubated medium and incubated in a 37 degrees Celsius, 5% CO2 humidity, saturated atmosphere overnight. At the very next day, the excessive mucus from the tissue was removed and 100 microliter exposed to the apical layer. And after the 24 hours incubation period, we done MTT. So this process follow for the DRF study. For the main study, we took a one step before the treatment, we also took the TR measurement, and after the treatment, we took the TR measurement. And for the main study, we took also the MTT, LDH, and histopathological examination. So these are the simple procedure we follow for the experiment. So now the result part. So first from the MTT in DRF. So here we can see by increasing the dose concentration level, the viability of the cell get decreased gradually. At the highest dose level of the 500 gram per liter, we hardly found any cells viable. Where the second dose, 10 gram per liter, we found 2% viable cells inside the insert. So all the dose for the main study selected below the 10 gram per liter concentration. So we selected 0.1, 0.25, 0.5, 1, 2.5 and 5 gram per liter concentration for the dose of the main study. So here are the results from the main study. Here we also see that as increasing the dose concentration level, the viability of the cell get decreased by increasing the concentration. At the highest concentration, we found that only 5% cells are viable inside the insert. And in the lowest concentration, the cells viability are get in a larger amount. So here we see the viability in the lowest concentration are more than 100%. This is due to the cell volume inside the insert are larger than the vehicle. So we move with the next endpoint, which is a tier measurement, the transepithelial electric resistance. So this is the instrument we use to measure the tier of the various group tissues. So here are the electric console which connected with the electric rod and which have the two electrode. So this is a placement. This is the placement of the electrode in a well plate. So it passes the electric resonance through the insert and collect the resistance which created by the cells. So here are the few results from the tier measurement. Here we also found out that by increasing the dose concentration, the tier resistance get decreased. This is due to the in higher concentration there are hardly any cells found viable. And for the positive control, we found the 38.13 resistance inside the insert. So moving ahead with the LDH. So as we know, all the living cells have the lactose dehydrogenase enzyme present inside it. When the cell get damaged or dead, they release the LDH. So higher concentration of LDH means there are more number of the cells are damaged or dead. So here we can also see by increasing the concentration, a higher value of the LDH we observed. At the 5 gram per liter concentration, the LDH value goes up to the 804. And at the lowest concentration, we hardly found any LDH. So this is confirmed our test reliability. So moving ahead with the next examination, we took the histopathology. So here are the few photographs from the control and vehicle control group. We can see all the cell structure are intact perfectly fine. Even we can see the ciliated body, the cell structure and membrane are completely intact together. Here are the photographs from the two low dose, 0.1 gram per liter and 0.25 gram per liter. So here we can see some of minor effects are created by the test item, which is the ciliated bodies are disturbed somehow. Some apoptic or necrotic cell found present and some membrane are get disturbed. So here the photograph with the two mid dose, 0.5 gram per liter and 1 gram per liter. Here we can see the more number of the apoptic or necrotic cell found present with comparison with the lower dose. Loss of the cell layer was observed, flattened epithelium was observed, and some of the membrane get disturbed. And here are the two highest dose, 2.5 gram per liter and 5 gram per liter. In this highest dose, we hardly found any cells present. Loss of cell layer was observed, and more number of necrotic hardly found any cells was observed. And these are the photographs from the positive control. We see the upper layer was get ruptured. More number of apoptic necrotic cells were found present. So here the conclusion. By comparing all three test data, we can say by increasing the concentration level, the cell viability get decreased, tier measurement get decreased, and high number of the LDH value was observed in a higher concentration. So by conclude this data, we can say that the LC50 of the lambda cyhalothrin 5% EC was found between the 1 to 2.5 gram per liter. Based on the severity of the parameter, the lowest observed adverse effect level of the test item was found to be 0.1 gram per liter. So these are the few team members who are involved in this research. The first person is Dr. Dheeraj, who take the histopathological examination. The second person is Mr. Jaideep, who took the LDH assay, and Dr. Vishwesh, myself, and Abhilasha, who conduct the overall research. So by completing the first step, our now inline project is on a epi-alveolar model, which we are going to conduct in earlier December. So this all from my side. Any questions?
Thank you, Mr. Jermish, for that very interesting and informative presentation. We are open to questions from the audience, anybody from the online. Can you hear that, Mr. Jermish, or should I repeat the question for you? Sir, can you use the mic?
It's not loud. Can you please read it out?
Yeah, 1 minute.
You have used formaldehyde. So my question is it the recommended standard or you just thought I should use it?
Actually, till date, there is no published data available. So by the knowing toxic property, we use the test item as well as the positive control.
Okay. So do you think that it's a proper or appropriate standard or some other kind of volatile material like maybe also considered? Any other alternative do you consider? Or you are satisfied with formaldehyde? How you maintain the concentration, that range of the concentrations of formaldehyde when you expose cells?
I think you use the what is it called? Lambda.
Sir, as a test item, we use lambda cyanogen 5% EC.
So that is your compound. I mean test compound. I mean I am just talking about formaldehyde. How you standardize doses for positive control? Yeah.
So we use the formaldehyde because formaldehyde is considered as a non-toxic. So, but in this case, we found that our test item at a higher concentration are more toxic than the positive control.
Dr. Raisuddin, if I get that, you're asking how do we determine, is the formaldehyde right control or something?
Yeah, how you standardize the dose which may be comparable to your test compound.
The dose ranging study for formaldehyde also.
No. In DRF, we use only the test item.
I think this was just taken because we know it crosses the cross-link and we know it will have damage. So along with that, he has done some other compounds. This one we know for sure it causes the damage, and that's why it was taken as control.
Like for AIMS test, there are recommended standards like EMS or hydrogen peroxide, these and that. But in this case, you use formaldehyde. Yes. But I got the answer. Thank you.
Thank you, sir.
Thank you, Mr. Jermish. We move on to our next speaker, Ms. Priyanka Mishra. She's a research officer at GRF Global and she's done her master's in microbiology from Ferguson College. She works as a study director for conducting in vitro HCLAT studies. She has experience in handling of different mammalian cell lines and in vitro assays like keratinocytes, DPRA cell cycle apoptosis, and HERG assays. Now I invite Priyanka Mishra to conduct the session on 3D spheroid models and its application in assessing long-term hepatotoxicity. Over to you, Priyanka.
Yes, good morning, everyone. So is my screen visible? Is my screen visible
Yes,
to all of you?
Very much, Priyanka.
Okay. So good morning, everyone. So today I'm here for presenting my research work on application of 3D spheroid models in assessment of long-term hepatotoxicity. So before we actually get to know what 3D spheroid models are and how have we used them for assessment of hepatotoxicity, let's first have a look at the origin of cell cultures. So the origin of cell cultures dates back to 1900s, the time when researchers actually identified that it is actually possible to isolate cells from an organism and culture them outside of their natural environment. So this typical method by which different types of cells they were forced to grow on a flat surface. This method typically became popular as 2D cell culturing. So this method of cell culturing became the method of choice since it was found to be both simple as well as convenient. But then it had certain disadvantages. Like these 2D culture systems, they were not able to recreate the physiologically relevant behaviors. Also, they are known to lack the tissue-specific architecture. And the basic point is the basic cell-to-cell communication is absent in 2D cell culture systems. So due to lack of all of these features, experimental findings generated using 2D culture systems may not be directly extrapolated to in vivo outcome of interest. Also, one important point is even in a human body, cells don't grow as 2D. And it is precisely the human body that one needs to model to develop better therapies against diseases. So that's the reason one should look in for a model which is both like having the features which are lacking in 2D and is also a more closer representative to the in vivo system. Something like 3D models, typically spheroid models. So what are spheroids? Spheroids are basically three-dimensional cellular aggregates which are formed when intercellular interactions are favored over attachment to a substrate. Now, the spheroid models, they are known to mimic the tissue microenvironment in a much better way than the 2D culture systems. When compacted, a spheroid typically is known to organize themselves into three distinct layers, with the outer proliferating zone, where the cells are very much proliferating and metabolically active. Followed by it is the quiescent zone, where the cells are metabolically active, but they are not proliferating. And at the center of the spheroid, you have the necrotic core, which is dense in terms of the metabolic waste. Now, why is spheroid considered an ideal model? Because it is known to promote cell-to-cell interactions in a much better way than as compared to the 2D cell culture systems. Now, what are the different areas where the spheroid models can be used? One of the most common application of spheroids is in studying drug-induced liver injury or long-term hepatotoxicity. So drug-induced liver injury, which is also known as DILI, it is one of the most common causes why drugs are withdrawn from the market after post-marketing. The second application is in wherein you can use cardiac spheroids, which mimic the human heart microenvironment, and you can study cardiotoxicity. The third application is in the areas of finding potential cancer therapeutics. Also, spheroids hold enormous potential in the areas of regenerative medicine and cell-based therapies. They have also been used in preclinical drug screening and validation when you are monitoring cellular responses to therapeutic interventions. Now, having known what are the different applications of spheroids, let's look at the different techniques by which spheroids can be generated. So there are multiple methods being mentioned over here, some of which are like use of ultra-low adherence plate, wherein the surface of such plates are being coated with a protein repellent material, because of which the cells don't attach to the surface. Instead, they form compact spheroid-like structures. Bioreactor method, wherein cells are under constant agitation, and because of which the cells don't sediment down and instead form spheroid-like structure. You have cell sheet engineering method of spheroid generation, wherein typically thermostable hydrogels are used and the temperature of which can be controlled for formation of spheroids. Pellet culture is one of the method wherein non-adherent cavities are being used for formation of spheroids, and such cavities are typically U or V bottom wells. And the most primitive method of spheroid generation is the hanging drop method, wherein spheroid generation is confined within the bottom of a droplet. So out of all of these methods which are being mentioned over here, at GRF, we have tried and tested two methods for spheroid generation, which are the hanging drop method and the pellet culture method. So first are the results of the hanging drop method. So as you can see here, we have got multiple spheroids in hanging drop, but since there are microliter of volumes being used for generation of spheroid, it's a bit laborious work. And during media exchanges, there are quite chances that you can lose the spheroid. And when such low volumes are being used, even evaporation rate is pretty high. And because of which you can, the cells can die, you can eventually lose the spheroids. So that's the reason we didn't proceed further with this method. And for all our experiments, we have performed spheroid generation by using the pellet culture method. So this is the typical schematic of how have we generated spheroids for our experiments. We have typically focused on forming hepatic spheroids because we wanted to study long-term hepatotoxicity. So for this, what we have done is we have used the hepatic lineage cell line, which is HepG2. So we have seeded the cells. After they have attained the required compactness, we have exposed them to the test chemicals, and at different time points, like after 24, 72 hours, 15 and 28 days of repeat exposure, we have done the hepatotoxicity assessment. Now, this is how our spheroid growth has been with respect to time. So as one can see over here, just after 24 hours of cell seeding, the cells are just aggregated together. They have not formed the typical spheroid-like structure. But upon increasing culture duration, one can clearly see the compactness the spheroids has attained. And gradually, also there is an increase in the spheroid size, which one can see. Now, this increase we have also quantified, and here are the results. Wherein on the x-axis, we have the culture duration in terms of days, and on the y-axis, we have the size in microns. One can clearly see here that the spheroid is increasing, gradually increasing in size upon increasing the culture duration. And the spheroids typically, they were close to 200 micron at day 3, and they have doubled in size upon this 4 weeks of culture duration. Then to visualize the cell layer organization in spheroid, we have sectioned the spheroids and have performed the H&E staining. And these are the representative images, wherein, as you can see, at the periphery of the spheroids, we can see a dense layer of proliferating cells. And as one moves towards the center of spheroid, the layer of proliferating cell is getting diminished. Next, to characterize our spheroids, we have chosen different markers and have performed the characterization using real-time gene expression technique. So for characterizing a spheroids, we have chosen different markers, like we have chosen a marker for proliferation, which is Ki67, and a marker, a pro-apoptotic marker, which is BCL2. So one can clearly see that as compared to monolayer cell, there is little or no change in the expression of Ki67 and BCL2 with respect to time. But in case of liver-specific markers, albumin and alpha-ketoprotein, there is a gradual increase in the expression of all of these markers when you compare it with the monolayer. And we have also selected CYPs and UGTs, which are involved in phase 1 and phase 2 metabolism. So even in case of CYPs, you can see that as compared to monolayer, there is enhanced upregulation of all of these markers being observed. Even in case of UGT, there is a gradual increase in the expression of UGTs with increasing duration of spheroid culture. So altogether, this results show that when spheroids, they are forced to grow in a 3D conformation, they develop into more metabolically competent state. So now that we had these results, we had the metabolically competent model, we thought of using this for our hepatotoxicity studies. So these were the 4 compounds that we have selected for our study, each belonging to different DILI classes. Acetaminophen, which is an antipyretic agent, it belongs to the low DILI. It causes low drug-induced liver injury. Diclofenac, which belongs to the NSAID category, it's a high DILI causing drug. Amiodarone, it belongs to the severe DILI concern class. And streptomycin, which is an antibiotic, its prolonged use is not known to cause any liver injury. So basically, throughout our research work, we have done 2 assessments. One is we have performed a comparative hepatotoxicity assessment. We have checked for the hepatotoxicity caused by all 4 compounds on 2D monolayer, and we have compared it with the 3D models. Next, we have also done the long-term hepatotoxicity using 3D spheroid models. So firstly, I'll be sharing with you all the results of the comparative study. So here are the representative images of hepatotoxicity caused by acetaminophen after 72 hours of treatment. So here the pink line is for the 2D monolayer cells which are being exposed to acetaminophen, and orange line is for the 3D spheroid cells. And on the x-axis, we have the concentration in micromolar, and on the y-axis, we have the viability. So one can clearly see upon exposing acetaminophen to the 2D monolayer cells, one can see toxicity up to the concentration of 6,200 micromolar, because of which the IC50 value was reported as close to 5,000 micromolar. Well, when the same compound was being exposed to three-dimensional setup, you can see toxicity up to the concentration of 12,500 micromolar with an IC50 value being reported as 15,000 micromolar. So we have done this toxicity assessment not just with one but all 4 compounds, and here is the table showing the IC50 values for both 2D as well as 3D setup for all 4 compounds and for the 2 time points, that is 24 hours and 72 hours. So for 24 hours time point, one can clearly see that except for amiodarone, which produces toxicity in 2D monolayer cells with an IC50 value of 48 micromolar, rest all the 3 compounds showed no signs of toxicity, and that's the reason the IC50 value was reported as greater than the highest test concentration. While after 72 hours of exposure, except for streptomycin, which was a non-toxic drug which didn't show any signs of toxicity, rest in all the 3 compounds we have got extreme toxicity with an IC50 values as mentioned in the table here. One thing to note is whatever IC50 values we have obtained for the test compounds in 2D, a 3 times higher concentration was reported as an IC50 value in 3D. So based on these results, we can say that okay, steroids are better at mimicking the physiological microenvironment, and they are also a better predictor of toxicity. So next are the results of long-term hepatotoxicity that we have performed using the 3D spheroid models. So here are the results of the spheroids after being exposed to the test compound streptomycin. So streptomycin, which is not known to cause any liver injury, its 24 hours exposure didn't cause any visible changes in the spheroid even at the highest test concentration of 5,000 micromolar. Similar was the response even after 72 hours of exposure when no morphological changes were being seen. But continued exposure up to 15 and 28 days caused morphological changes at the highest test concentration of 5,000 micromolar, but then no visual signs of toxicity were being seen. Next were the results of amiodarone, which is a severe daily causing drug. And since it's a severe daily causing drug, even 24 hours of exposure caused morphological changes at the highest test concentration of 125 micromolar. And upon 72 hours, 15 days, and 28 days of repeat exposure, complete toxicity was being observed as the highest test concentration, and a gradual reduction in the spheroid size was also being observed. This proves the extreme toxic nature of amiodarone. Now, next is we have also quantified the change in the spheroid size upon treatment with the test chemical, and here are the results of the same. So the first graph here is of streptomycin, wherein on the x-axis we have the concentration in increasing order, and on the y-axis we have the spheroid size in micron. So since streptomycin is presumably, it's assumed to be a non-toxic drug, its exposure to the spheroids, it is not causing any inhibitory effect on the spheroid size. Like one can clearly see whatever size the spheroids had at 24 hours, even after 28 days of repeat dosing, the spheroid size have gradually increased and it has not decreased. But in case of acetaminophen, one can clearly see that at the toxic concentrations, right from 25,000 to 3,000 micromolar, whatever size was when we were exposing the spheroids with after 24 hours at 24 hours, the size has gradually reduced after 28 days of repeat exposure. And similar were the responses obtained in case of diclofenac and amiodarone, wherein the toxic concentrations were reducing the size of the spheroids in much significantly. So next are the hepatotoxicity results for all 4 compounds. So here are the results of streptomycin, which is a non-daily drug. So basically streptomycin exposure, as one can clearly see, it didn't cause any greater than 50% toxicity even after 28 days of repeat exposure. The viability at all the concentration was above 50%. And that's the reason for all the time points we have reported IC50 value as greater than 5,000 micromolar, which is the highest test concentration to which the spheroids were exposed. Next are the results of acetaminophen. So in case of acetaminophen, 24 hours of exposure didn't cause any toxicity greater than 50%, even at the highest test concentration. But upon 72 hours of exposure, a complete cell death was being observed at the highest test concentration. And even at the second concentration of 12,500, there was a bit of toxicity, close to 50% being observed, because of which the IC50 value was reported as close to 12,000 micromolar. And upon 15 days and 28 days of repeat exposure, complete toxicity was being observed up to the concentration of 1,500 micromolar. And based on the results of this, the IC50 value for these 2 time points was being reported as 1,300 micromolar for 15 days and 800 micromolar for 28 days. Next are the results of diclofenac, which is a high DLE causing drug. So exposure of this drug to 24 hours to steroids had no signs of toxicity. But upon 72 hours of exposure, we've got an IC50 value close to 600 micromolar. And upon 15 days of exposure, one can clearly see that we have got toxicity greater than 50% at till the concentration of 250 micromolar. And similar were the responses even after 28 days of exposure. Therefore, the IC50 values were for 72 hours was being reported as 600 micromolar, and for 15 and 28 days, they were reported close to 150 and 120 micromolar respectively. Next are the results of the severe liver injury causing drug, which is amiodarone. So its exposure to steroids for 24 hours had no greater than 50% toxicity, because of which the IC50 value for this particular time point was reported as greater than 125 micromolar. But upon 72 hours of exposure, complete cell death was being observed up to the concentration of 62.5 micromolar with an IC50 value close to 30 micromolar. And upon 15 and 28 days of repeat exposure, one can clearly see a decline in the number of viable cells up to the concentration of 15.63 micromolar. And that's the reason the IC50 value for this compound was being reported as close to 11 micromolar. So it's indeed that the severe liver injury causing drug amiodarone is known to be toxic even at the lower concentrations of 15 micromolar. Next, one of the parameter LDH release, we have also checked for this, which is also an indirect measurement for the cell viability. So this assessment we have done after 15 days of exposure, and here are the results. So one can clearly see here from the results that at the highest test concentration for all the three compounds, we have observed maximal LDH activity, and upon reducing the concentration, even the LDH activity was found to be reduced. And in case of acetaminophen, surprisingly, we didn't got any LDH release. So finally, what do we conclude from our study? That steroids basically is a reliable model for assessment of long-term hepatotoxicity, a feature which is lacking in 2D. Because they are in 2D, the cells are highly proliferating. That's the reason hepatotoxicity for a long period of time cannot be performed with using those cells. So steroid proves to be an ideal model in such a case. And steroid or any other 3D model can be helpful in bridging the gap between 2D cell culture and in vivo assays. And also such models can improve the predictive power of in vitro drug screening. Having said that, I've already shown the results of long-term hepatotoxicity with you. I've shared the results with you. But with using such steroid models, we have also performed assays like the non-alcoholic steatohepatitis and CYP induction studies. So non-alcoholic steatohepatitis is a fatty liver disease for which up till now no approved therapies are being available in the market. So with steroids, we have also done assessment of those assays, and we have got promising results. But then due to time limitation, I was not able to share the results with you. So this was all with respect to steroids, and what we have, what we are planning to do in future is, whatever experiments we have done up till now was with the static model of steroids. So we have thought that integrating fluidic system with our 3D models can help in, mimicking the physiological environment in a much better way, and the results obtained with such model will be even more reliable. So yes, that is it from my side. Thank you.
Thank you, Priyanka Mishra, for a wonderful presentation on how 3D models can be better predictive models for long-term hepatotoxicity studies. We are now open to questions from Ms. Priyanka. Anybody from the audience would like to ask a question? Anybody online? Okay, Priyanka, I may have one or two questions for you. If you can you just go back to the slides where you have shown the IC50 values of 2D versus 3D models?
Yes, ma'am.
Yeah, so over here in your comparison of 2D versus 3D, we see that 3D models have a very high IC50 value. Your conclusion was that 3D models are better predictive models for hepatotoxicity. Can you justify how a higher IC50 value can be a better model as compared to a lower IC50 value?
Ma'am, as I said during my introduction slides, like even in our human body, the cells are not organized in a 2D, like they're not in a flat structure, right? They are organized in a three-dimensional state. So for any drug to penetrate inside such, in 2D, whatever information we are getting is when you're exposing compounds directly on the monolayer. But the organization in our body is not in that way. So typically the results with 3D are better at mimicking what is happening in your body. So that's the reason I made a statement that assessment of hepatotoxicity with spheroid is better, because as per in our human body, how the cells are organized, with 3D models, whatever values we are getting, so that will be a more closer representative to the in vivo system. So that's the reason I made that statement.
Okay. So in that case, what you mean to basically say is 3D models, 3D spheroids replicate the in vivo conditions to a greater extent. Then in that
Yes.
situation, have you shown the drug delivery mechanism within a 3D model is very similar to that of the human body? Any studies on that, if they have been done? Because what I believe is, since 3D spheroids are a very compact and an integrated structure, drug penetration and drug exposure can be a difficult thing at low concentrations. So they may
Right.
not be able to really replicate the in vivo conditions. This is what my understanding of 3D spheroids are. But looking at the data, since the IC50 values are very, very high, almost like 3 times higher than a 2D system, how would you
Yes.
justify this, that there is better drug penetration in 3D models, or better drug exposure, or better contact of the drug which is replicating to the human body? How do you think you can justify that?
Right. Regarding drug assessment, like any studies with regards to how the drug penetration mechanism is, we have up till now we have not done any such studies. But then yes, that is a good point. One can think of such things. And we can think of this particular aspect for our future work.
Okay. Yeah, because your streptomycin studies shows very similar results on a 2D and a 3D. If that would have been the case, there would have been certain level of toxicity with streptomycin as well on a 3D spheroid model, since it's a drug at the end of the day. So I was just wondering, since there is good replication of 2D versus 3D on the streptomycin model, how come in the higher drugs, which are more, say, hepatotoxic, there is such a stark variation between the two models? So just to find out, so in case of future, if you do those studies, please do get in touch with me. I'd like to know what has happened as the outcome of 2D versus 3D. Yeah. Thank you so much for clarifying all the questions. Now, yes, sir, please.
Yeah, I was listening to the talk, and more importantly, the discussion between the chairperson and presenter. I would like to make an observation about the IC50 concentration, which is several times higher in the 3D model compared to the 2D model. And I appreciate Dr. Pradiksha's point that the 3D model should have more efficiency in responding to the drugs and chemicals than the 2D model. And more importantly, a point which was not mentioned in your discussion was that one of the objectives of the 3D model is to have the intercellular junctional complexes to develop, which you don't get in the 2D model. So you have tight junctions, gap junctions, all kinds of intercellular junction complexes will develop, and there will be transporters. In the 3D model, transporters are also very clearly established. The three-dimensionality of each cell is decided, top, side, bottom. So these transporters and the intercellular junctions are better contributors towards physiological performance and responding to drugs and the toxicants, whatever be. So as Pradiksha said, you should investigate further. As you have also said that you will do it from these perspectives, 3D perspective of intercellular junctions and junction complexes and the transporters. be investigated, and we may have a presentation in our next conference or on our next meeting. All the best.
Thank you.
That was very valid point by Dr. Akbar Shah. I think Dr. Abhay Deshpande has raised his hand and would like to comment. Can we have Dr. Abhay Deshpande please come in?
Thank you, Dr. Pratiksha. I am speaking more as a scientist than JRFian here. One point that I would like to make with respect to the 2D versus 3D observation. One is because of the limitation of number of cells, the contact inhibition in place, the expectation of higher number of cells on the surface of a spheroid, primarily because you have much more surface, you're likely to see more death. That's one part of the story. Second is the significance of the 3D spheroid, the detection of LC50 with 3D spheroid is more realistic than the one with 2D, primarily because as you can see in 2D, the prediction value for IC50 is much less as compared to that, whereas the human exposure is likely to be more for the cells. Having said that, some of the limitations of such studies are because, as I spoke yesterday, we are representing only one cell. Now, liver per se is not one cell. You have 4 or 5 other cells. You have Kupffer cells, you have stellate cells, you have the endothelial cells, and so on and so forth. So, truly speaking, the representation of IC50 is not as good as you would like it to be, but it's much better than what you would have in a monolayer. So that's what are my thoughts. And Dr. Akbar Shah, would you like to express your views on that?
No, sir's okay with your comments. Thank you so much, Dr. Deshpande, for clarifying.
Thank you.
Thoughts on the IC50 value. So we'd like to now move ahead with the last speaker for the day, Mr. I.M. Barad. I'm privileged to introduce Mr. I.M. Barad as the Senior Research Associate at JRF Global. He's earned his Master's in Science and Biotechnology. Mr. Barad has emerged as a trailblazer in his field. Currently, he's serving as the Study Director at the JRF Global, and his role extends far beyond his mere title. His expertise lies in the meticulous design and execution of various in vitro and genotoxicity studies, adhering rigorously to the OECD guidelines and the ISO guidelines. His proficiency spans a range of critical assessments, including cytotoxicity tests and in vitro and in vivo studies, such as MMT, HBRT, and HLCA, and many more. Mr. Barad's proficiency is in navigating the complexities of genotoxicity studies, making him a true asset to the research community. Today, we have the honor of welcoming Sir to the discussions of JRF's participation in the OECD TG428 study ring trial with 14C caffeine and its comparison with in-house GLP data. Over to you, Mr. Barad.
Thank you so much for the introduction. Is my slide visible?
Yes, sir, it's visible.
Okay. So, good afternoon, everyone. Today, I am here to present the JRF's participation in OECD test guideline 428 study ring trial, that is in vitro dermal absorption test using radiolabeled caffeine and its comparison with in-house GLP data. So we will go through the contents in which we will see the data of in-house validation as well as results of both ring trials, and we will compare those results with in-house data. So what is dermal absorption test? So diffusion of test item from outer surface of the skin in the receptor fluid. The receptor chamber was supporting grid and digest skin after tape stripping, except first two skin strips. So if we basically, if the test item is applied on the skin, it is measured that how much amount of test item is passing through the skin and how much amount is present in the skin. That is called as dermal absorption. So we have a dedicated facility, radioisotope tracer facility for performing dermal absorption assay, in which we have the major instrument is the flow-through diffusion system and the liquid scintillation analyzer, from which we can analyze the radiolabel material concentration. So in the dermal absorption study, first of all, we select the skin based on integrity test. In integrity test, we check the skins for their appropriateness. So if the skin donor has passed, then we select those skins for the study, and then we prepared that test item based on the experimental needs. We can use a radiolabel compound as well as on unavailability of radiolabel compound, we can also use as a cold compound. So if we are using radiolabel compound, we will finally analyze it on liquid scintillation counter. And if it is a cold compound, we will analyze it on LCMS as well as HPLC, whatever experimental need is there. So after selecting skin, we will place the skin in flow-through diffusion cell. This is the cell in which there are two parts. This is the donor cell, donor chamber, and the below this is the receptor chamber. This one. So the skin is placed in between both the chambers, and this chamber is placed on this fraction collector head, from beneath which the receptor fluid is passing. So when we apply the test item from the donor chamber, whatever amount of test item is passing through the skin, it will be collected in the receptor fluid below the receptor chamber, and the timely samples will be collected in the fraction collector. So based on experimental requirements, we will remove the test item by washing at particular time points that may be normally around 8 to 12 hours of time points. And after that, we will terminate the experiment at by washing the skin again at 24 hours. And after 24 hours, we will dismantle the diffusion cell, and the skin will be dried. And next step will be the tape stripping. In tape stripping, we use a particular tape, we stick it on the exposed part of the skin and remove it. In this way, we use 15 tape strips for each skin and each skin, and we analyze it afterwards. After that, the skin is digested. It is also used for analyzing the available material inside the skin. And after that, we will analyze all the collected sample in liquid scintillator counter. So during the in-house GLP validation, we have tested benzoic acid, caffeine, and testosterone using rat and human skin. So here is the image of skin after the tape stripping, so that we can here see the impact of tape stripping. Here we can see the stratum corneum is intact, while after the tape stripping, the stratum corneum layer is completely removed. So here we can see the how much efficient is the tape stripping. So here is the result of our in-house GLP validation for all the compounds: benzoic acid, caffeine, and testosterone. Almost a similar pattern was observed in the absorption profile. This absorption is after 24 hours of absorption into the skin in the receptor fluid. Here is the cumulative absorption which we have obtained after 24 hours of collection in the receptor fluid. We can here also see the almost similar kind of patterns in absorption, except we have in benzoic acid, the initial absorption was quite fast than other two compounds. So this is the design of the first ring trial which was conducted, and I'm proud to say that JRF is also part of this first ring trial and also the upcoming second ring trial. So in the first ring trial, 12 laboratories were selected for one run using the testosterone, and all the laboratories have performed these studies using their available applications, skin application area, skin origins, and diffusion cell types using the receptor fluid. So here are the results of first ring trial. We can see for the parameters like mass balance, absorption in receptor fluid after 24 hours, and potentially absorbed dose. That is absorption in receptor fluid plus skin membrane and all tip strips except the first two tip strips. So here we can see the results of JRF internal validation for testosterone were quite similar to the results that were obtained in the first ring trial. That is, recovery was 95%. The absorption in receptor fluids was around 3.8%, and the potentially absorbed dose was around 8%. So here we can see that the variation between laboratories was not that much consistent. There was some kind of variation in the first ring trial. So to overcome that variation, second ring trial was conducted with the main objectives like to harmonize and reduce the potential source of variation, to identify the factors that account for the dermal absorption results variability, and also to demonstrate the reproducibility and repeatability of the results. So in the second ring trials, 6 laboratories were selected based on the similar apparatus available, and all the laboratories conducted 2 independent runs. So during the second ring trial, skin donors were distributed in manner so that inter and intra-laboratory repeatability can be demonstrated. So to do that, all laboratories conducted 2 runs and 2 donors per experiment were used. And for inter-lab repeatability comparisons, each skin donor were distributed in 3 laboratories, which we can see how it was distributed. And in each donor is used in duplicate per experiment, and total 8 diffusion cells were included in each run. So to standardize all the experimental step, standardized protocol was provided for each step, and the main thing, the skin was supplied by common donor, and the skin was from abdominal region of having appropriate thickness and appropriate diameters. Also, the experimental consumables were standardized by all. So all the labs used the consumables from same supplier, product number, and lot number. Also, the dose concentration used for caffeine was 4 mg per ml, and the vehicle used was PBS. So the skin samples were provided by the same supplier to all the labs during this time period. And these are the results of the second ring trial conducted for caffeine for the parameters like maximal flux, absorption in receptor fluid, and the potentially absorbed dose. Here we can see that all the results for all the labs were very consistent to inter-laboratory as well as intra-laboratory. So also for the results of GRF internal validations were quite comparable with all the results obtained in the ring trial 2. Maximal flux was 0.18%. The absorption after 24 hours in receptor fluid was around 5%, and the potentially absorbed dose was 5.8%. The cumulative absorption obtained between all the labs was almost also consistent. So the mass balance and mean recovery, which was obtained from the ring trial 2, was around 99.10%, and also the GRF internal validation results was 97%. And we can also see here that the mass balance obtained from all laboratories was quite comparable, and the distribution profile, that is recovery in receptor fluid and tape strips, was almost consistent in all the laboratories and in all the experiments. So when we compare the results of ring trial 1 and 2, we can directly see that the results were very consistent in ring trial 2, as there were many standardization ways were considered. So here, one thing is to note that the lab designation A2L and A2F do not correspond to each other. For example, the lab A in ring trial 2 is lab I in the ring trial 1. Although the results of the lab A in ring trial 2 were a bit higher than other labs, the historical data which was therefore the lab A was quite in line with all the laboratory results obtained for caffeine in ring trial 2. So from this ring trial, there was low intra-laboratory variability in all the 6 laboratories across 2 experimental repeats. Also, caffeine recovery was similar between laboratories, that is 99%, and a low inter-laboratory variability was observed in 5 out of 6 laboratories. That is a mean caffeine absorption of 3.9%, and the absorption in laboratory A was higher than other labs. It was considered that the laboratories earlier participation data and data that the variation was considered to be associated with an accidental COVID-19 pandemic related delay in the sample shipment. The skin associated factors other than integrity, that is in this case the delay in sample shipment, may also affect the absorption and variation during the dermal absorption study. So understanding the variability in the in vitro dermal absorption assay with human skin will continue contributing to the design and the conduct of appropriate regulatory studies and adding further considerations to the existing OECD dermal guidance notes. The ring trial demonstrated that OECD test guideline 428 assay, that is in vitro dermal absorption test, can robustly and reliably performed by different laboratories. The results from GRF internal validation experiments, the mean caffeine absorption was able to replicate similar conditions. This is the article from which the results of ring trial were. It can be referred if anyone is having any kind of anyone wants to know any kind of things about this assay. So these are the reference which we have used to conduct this assay, and that is all from my side. Thank you for your attention, and I would be happy to answer any questions.
Thank you, Mr. Marat, for that extensive presentation. We are open for questions from the audience and from people online. Anybody? Okay, so that brings us to the end of this session, a very interactive session of J Research Foundation and the team back in India, working very, very strongly in silico models for computational chemistry and mutagenicity predictions, development of 3D spheroids for hepatotoxicity, indigenous RHE skin models for evaluation of skin sensitization and irritation studies, in vitro inhalation toxicity assays, and participating in the prestigious OECD for validation studies. I believe that GRF is doing an extensive, integrated, and a wholehearted approach towards working with their objectives of alternatives to animal testing. And I, on behalf of all the organizing members and the people present at this conference, would like to wish you all the very best for your future endeavors. And thank you very much, everyone, for giving a patient listening to this session and being participant to this conference. My heartfelt gratitude to all the organizing members of this conference and the event, the people who have been at the forefront and at the back for making this conference a big success. Thank you, Dr. Rahman, for inviting me as a chairperson for this conference. I'm looking forward for many such associations and working with you all in the future. Thank you so much. Have a great day.
Thank you.
Thank you. Thank you. Thank you.
Thank you, ma'am. Thank you, speakers, for your insightful session. Now I request Professor Dr. Syed Ziaur Rahman, sir, to hand over the token of appreciation to Dr. Pratiksha Aligarh, ma'am.
Now I would like to request all our guests to join us for a short and refreshing tea break. We will resume our next session at 12 o'clock. But before that, I would like to request our honored judges, Dr. Pratiksha Alag ma'am, Ms. Savita Nutan ma'am, and Dr. Ankita Pandey ma'am, to kindly score the posters which have been screened and selected. And I would also like to request the guests, while having the tea break, please go and see the sheer efforts being put into making the posters and appreciate it. Thank you.
Distinguished delegates, esteemed guests, ladies and gentlemen, good afternoon to all. I'm Dr. Anand.
I'm Dr. Swati, Junior Resident, Department of Pharmacology, JNMC, AMU. It gives us great pleasure to oversee this session of the conference.
For this session, I kindly welcome Dr. Akbar Shah, President of SAAE, and Ms. Savita Nudan ma'am to be the chairpersons of this session.
Ms. Savita Nutan ma'am, founder and CEO, Medicine Without Cruelty, Pool, England, a dedicated educator and former scientist who is deeply committed to advancing animal-free medicine research. With a decade of experience in the medical sciences, ma'am has witnessed the limitations of animal testing in medical research firsthand. Ma'am's mission is to raise awareness and promote innovative, cruelty-free technologies that can revolutionize the field.
I now request Dr. Akbar Shah sir to introduce our distinguished guest and the speaker for this session, Dr. Christian Pelavoisin sir, who will be available online. Sir will be delivering a speech on 20 years of revolution in toxicology driven by 3D models.
Christian, good morning. Nice to see you on the screen. I think it was after a long time. We are at least see this way after we met in Korea, right? So that's nice. Just to introduce that we are personally known to each other for several years, and so then personal inquiry. I do the official business of introducing Dr. Christian to the audience. So Dr. Christian Pelvoisen is a distinguished scientist, scientific director at Matrix Life Sciences. You know, Matrix Life Sciences, the big companies in 3D tissue culture. Like Episkin, we also do the reconstructed human epidermis. Matrix also is a tissue engineering company, and like Christian is the scientific director. So with a rich background of toxicology, biotechnology, cosmetics, and medical device industries, skilled in research and development, toxicology, and in vitro assays, Pelvoisen has been instrumental in developing innovative reconstructed human tissue models and in vitro models for assigning product safety across diverse industries, including cosmetics, medical devices, and pharmaceuticals. His unwavering commitment to advancing alternative methods of animal experimentation and his role in developing, validating, and standardizing in vitro techniques make him a pivotal figure in the field. Additionally, his dedication to knowledge sharing and education, particularly in the realm of predictive toxicology, further cements his status as a leading expert in the industry. So having done the job of reading out his brief biography, I should, I am here, I should say a few words about his contribution to the society and to India. Dr. Christian is personally and technically known to me for more than a decade, as I am one who is spearheading the movement of alternatives in India. Dr. Christian, representing at that time a big company, another company of the L'Oreal, the Episkin, he got associated with me through my contacts, and he was in fact participating in quite a few number of workshops, number of training workshops in India. And it is a very, I should very particularly make a statement that these are training workshops which Christian used to organize without expecting any travel assistance from the organizer, as namely the society and my earlier center, and without even claiming any cost of the tissue which he would bring for the training. He is an expert in reconstructed human epidermis, in addition to the several other things. So it is this product of the Episkin, formerly and now Matrix, Christian has been bringing to India on his own and engaging in the training activity. And at least 2 training activities are conducted in Mumbai, the Pradikshas Institute. And at my Mahatma Gandhi Door and Camp Center, he came 3-4 times. And at that time, India, the DCGI and the regulatory authorities banned cosmetic testing in animals, and we didn't have an alternative. So we quickly got into touch with Christian. He was so nice. He brought the samples to India, and then he demonstrated the application of the sample in cosmetic testing. And now people are a big relief. They know that there is an alternative. OECD approved alternative is available for cosmetic testing with animals. And the credit in a very big way goes to Christian and to a certain extent Episkin informally and Matrix now. And he ought to have come in person, and it's very, very, very bad and tragic thing happened when he was coming. And he had a little problem with his passport when he was almost checking in at the airport in Paris, and he was not allowed to board the flight for this technical reason, and he was asked to go home. So it's a very sad impression, he went back and he shared his agony rather through mails. So we joined him in his displeasure, distress, and but in spite of it, he has been present for this thing. Another thing which I should, of course, announce here is that when he was in Episkin, he was very generous to motivate the Episkin company to help us with the best poster and best oral presentation awards in the form of gold medals. That's big money. 4 grams, 3 grams, 2 grams, and 1 gram for second and third prizes. That was Episkin money with Christian brought. And now that he moved over to a new company, Matek, he still volunteered to say that he would continue to support the award contribution. And he sought how much money we would require, and we gave a suggestion. And he was very generous to transfer the money so quickly. And the award which should be presented this evening, the verdict session, the money actually comes from the Matek company at the initiative of Dr. Christian. So I should very specially thank Christian for this, generally for all the things we have been doing to India, to the Mahatma Gandhi Center, and to the society. I should also very specially thank you for this very nice gesture of finding money for presenting the awards for the students. And with these few words, I invite you, Christian, to make presentation of the topic and follow with the discussion. Thank you so much.
Thank you very much, Dr. Agbarsha. Thanks a lot for this kind introduction. Thanks to the organizer for the invitation. Apologize not being with you. It's really a pity, but effectively, I had a trouble with my passport. In any case, I'm very happy to be there because I follow the evolution of the Society for Alternative to Animal Experiment in India. I follow the evolution of the ecosystem in India. And it's really exciting for me, and that's why I proposed this talk about having a look on what we need to do for replacing the animal, but also to have a look on what has been done. Sometimes we always say, okay, we are far from replacing the animal everywhere. There are so many things to do, so many money to put on research. It's true. But in the same time, a lot have been done. And this is what I would like to show to you, is that we have done a lot have been done, thanks to 3D models. Not only, but 3D are very useful for replacing the animal. And it's a short overview that I want to give to you on this. First of all, animal experiment is a long history for humanity, because as soon as the antiquity, we can see that some experiment, vivisection, dissection have been performed. And even if during all the Middle Age, no science have been really done, as soon as the Renaissance in the 17th, 15th and 17th century, we can see that animals were used more and more to start to better understand the physiology of the animal, of the human, and in the 20th century, 19th, 20th century, with the modern revolution, we observed a huge explosion of the use of the animal for science, for education, for toxicology, etc. Up to this date is important. You know it, in 1959, Russell and Burke, who started to build the concept of alternative. They were the first really to have a scientific approach of the subject, saying and edicting the free concept of the free air, reduction, replacement, and refinement. And from this date, we observed a slow decrease. Estimating the number of animal use in science in general is not easy, because they are not real statistic. And in this publication in 2015, tried by data mining from publication, to made an estimation, and they proposed the number of nearly 200 million of animal use every year. So it's a very huge quantity of animal each year that are used for different purposes. And this purpose can be for research, obviously, to discover a new treatment, to understand some pathology, to discover a new mechanism, physiological mechanism. But it can be used also for regulatory testing, for education. Education use also a lot of animal. And we have also a part not so negligible with all the quality control in pharma industry for each batches of new antibodies or something like this, where you need to check some quality of the batch. Today, I will focus on regulatory toxicology, the use of animal and the replacement of animal. What I will say is specifically for tox, but could be also extended to other domain in some situation. So why do we need regulatory toxicological testing? Because we can imagine that we can rely on the seriously of the industry to put on the market safe product, and we don't need to check. This is an ideal world, but in fact, we need to be careful. And this just an example. We can find several examples, but I find this quite funny because now when we have a look to this, we said that it's impossible that we do the same now. But I'm not sure sometimes. This was an exposition done by FDA in the 1932 that have been called by the journalist newspaper, the American Chamber of Horror. In fact, in the 30s, the regulation was quite simple and light, and it was really easy to put on the market new products. And these are some examples. For example, we have a lash lure product, which was a cosmetic eyelash dye that contained aniline, eye sensitizer ingredients that lead to a lot of allergy and blindness of some people. Some example here is a depilatory agent containing thallium acetate, which is a rodenticide, and lead to some total blindness and permanent hair loss of some users. We have the example for here of skin pigmentation product to treat some to remove the brown spot and to lighten the skin. And the active was mercury, and we know that it's really dangerous for the health of, but it was possible to put it on the market. And this is really, we can say funny, but sometimes it's not funny, the good word, but it was all a series of products containing for the skin, some gel and cream for the skin, and also some energy drink that contained thallium and radium, highly radioactive product that lead to the death of some people. So we can see that without rules, without law, without control, it could be really dangerous for the population. And that's why from this stage, several laws have been enacted in the world to reinforce the control of a product before customers can use it. And this is the regulatory toxicology. And the point is to assess two big effects, what we call the local effect, the effect observed where the product come in contact with the body, and the systemic effect, when the product can go inside the blood circulation and is distributed all over the body. For local effect, there are a series of endpoints to assess, quite described, quite well described. You can have corrosion, irritation, photoirritation, sensitization, photosensitization, absorption, metabolism, especially at skin level. This concerns a number of tissues, organs, limited. It's the eye, the skin, the gastrointestinal system, the respiratory system, all the tissue that are in the interface between the body and the external part environment. As soon as the product is able to go through this barrier, this mucosal, this epidermal barrier, they go in contact with the blood circulation, and we are in the situation of a systemic effect. We need to assess different endpoints. And in this case, we can see that it's much more complex because when we speak about systemics, it's nearly 80 different organs and different effects, the acute tox, the repeated dose toxicology that with different length of exposure, reprotox, carcinogenicity, toxicokinetics. So very complex endpoint to assess. But basically, this is what we have for toxicology to assess. And the gold standard for these toxicological endpoints are often the OECD test guideline, even if they are other standards and other guidelines to assess. But OECD is really a repository. It concerns a lot of countries, 35 countries in the world, and it's extended thanks to mutual acceptance of data to more than 50 different countries, where you have some test guideline, you have some GLP practice, and if you follow both, you can your test can be recognized everywhere in the world. Historically, the OECD have been created in the 70s. The test guideline for chemical started to be released in the 70s, 80s. Nearly all the testing were based on animal. And following the 3R principle, a lot of test guidelines have been refined to reduce the number of animals, to reduce the suffering of the animal, but it was very complex to replace the animal testing. And the first big change was achieved thanks to the 3D technology, and especially the 3D skin technology, which started in the 70s in the US and in France by rebuilding reconstructed human epidermis that allowed to start to develop some toxicological method for skin corrosion, skin irritation directly on these 3D models in vitro rather than doing in the rabbit. Another big step was the capacity to have an industrial production of these 3D models, because as soon as we speak about regulatory, it means that we need to have a very highly reproducible, standardized production of these models, 3D models. Thanks to this industrialization, the 3D models start to be a consumable for the labs. Any labs in the world can order the 3D models, and it's a consumable as another consumable used in a laboratory. Matek, founded in 1985, put on the market the epiderm model, which is a reconstructed human epidermis, in 1993. It means that these models, this technology exists for exactly 30 years this year. So a long history about this. The technology in tissue engineering, it's true for skin, but it's true for other models, is always based on the use of human primary cells. The point is really to have model mimicking the human situation. So we try always to access to primary human cells coming from donor to isolate the cells of interest. It could be keratinocyte, but it could be hepatocyte, it could be enterocyte, depending of the organ, to do cell banking, and after, in the good condition, with a good medium of culture, to be able in few days up to few weeks to reproduce a living organ in a plastic insert that can be used really easily in a laboratory. Here you can see what it looks like. These are some plates, 24 well plates with reconstructed epidermis, the epiderm model. The big interest is the air liquid contact. You can see that the surface of your reconstructed epidermis is in contact with air exactly as the human tissues, and is exactly the same for the other tissues, gastrointestinal, corneal, oral, respiratory. We have the same situation. We have the environment and the body on the other side. This allows to imagine a lot of experiments, and thanks to primary cells, we are really close to human situation, and also we have no limitation in the applicability. It means that you can apply liquid, solid, gel, cream, but also gas and particulate as you want. This is very important to develop a toxicological method. So thanks to this technology, the first success, big success in terms of animal replacement concerned the skin corrosion and irritation test. This test has been developed in 1944 by Dres in the US. It's based on the use on albinos rabbit. You shave a rabbit, you apply the product to test between 3 minutes up to 4 hours, and after you have a look to the skin, and you give a score based on the redness, the erythema, and the edema to be able to classify your product as a corrosive or irritant. And the big change between corrosion and irritant is the reversibility. It's not. It's a test that has been used massively in the world, using millions of animals during the last decades. And even if in 2D cell culture, some methods have been developed, this method was not able to replace this animal. It was only informative in parallel, but it was not possible to replace. This an example of what look like the Dres rapid test would be only a slight irritation up to highly corrosive, so it's really suffering for the animal. And not only the trouble of ethics about some this test, but there were also some trouble and question about the predictivity to human, because this method has never been validated, and we are not sure that it predicts 100% the situation of a human. But in my mind, the most important trickiness of this assay is the reproducibility. As it's a scoring system, depending of who is doing the test, you can have different results. And to this point of view, this recent publication is very interesting, because they collected from 1,000 different chemicals that have been tested several times. So the same chemical tested several times, and after they compared the result. If we have a look to the GHS category for skin corrosion irritation, you have category 1, which is a corrosive product, category 2, which is an irritant product, category 3, it's a mild irritant, and non-category, it's a non-irritant, in fact. And they have compared, when you have a product tested as a category 1, if the same product has been tested a second time, or third time, or sometimes they have been tested 10 different times, they compared the second results. And you can see that for extreme category, corrosive, it's quite reproductive. Each time you have a corrosive, you have good chance that if you do the same test the second time, it's also corrosive. Idem for non-corrosive, non-irritant, no category. But as soon as you go to in between category, especially for irritancy and mild irritancy, then you can see that it's not really reproducible. For product classified category irritant, it can be in a second assay classified as non-irritant up to 20% of the situation, or corrosive. And for mild irritant, it's worse. You have 1 chance on 2 to have different results. So that's why there were a need also for more predictive, more reproducible alternative method than the animal. Different methods have been developed, but the only method that has been validated as a full replacement of the animal was based on a reconstructed human epidermis. This is the test guideline 431 for skin corrosion. released in 2004 at OECD level, and it was the first time for OECD that they published a method, in vitro method, able to fully replace the animal. And few years after, we had the same principle, the skin irritation in vitro method in 2009. And if we have a look to the situation, and that's why sometimes it's good to have a look backward, we can see that it's really a success. In 2000, all the submitted and published data for skin irritation and corrosion were based on OECD test guideline 404 on rabbit. But since the release of the test guideline 431 and for the irritation, in vitro irritation, we can see a total shift from vivo, and now it's only based on in vitro. So it means that it's possible to replace the animal thanks to in vitro, and especially thanks to 3D models. The second essay, where we had also a big success, was the eye irritation. Again, albinos rabbit. You fix the head of the rabbit and you put in the conjunctival sac the product to test, a liquid, or could be also some solid. So it's it could be very suffering for method for the animal. And again, you assess, you score some effect, and you can assess the reversibility. It was more complex than skin because it concerned not only the cornea, but also the conjunctiva, the iris. So it was complex really to replace, and it took more time. But again, for this essay, we can question the predictivity for human, the ethical, there are no question about ethics for this essay, but the reproducibility also. And again, another publication from 2016, they downloaded data from the rich EK database in Europe, and they mined this data. And again, they compared for chemicals tested more than one time. They compared the result when you test one time and second time. And again, for extreme situation, it's not so bad. 70% of reproducibility for category 1, highly irritant. No category is very predictive, but for the intermediate category, when we have a look, when you have a 2A, for example, a mild irritant, a slight irritant, you can see that if you test in different several times after, you can have a no category or even a corrosive. So it's really not reproducible. And this is really questioning about the pertinence of the animal. So again, several in vitro methods have been developed. None were full replacement. The test guideline 492 is based on human reconstructed cornea. In this situation of 3D models of a human cornea, it's not a full replacement, but what is very interesting is that it's a method which is recommended by OECD when you think your product is no category. So it means that if you think that your product is not irritant, irritant, you can go with this method and you can classify it directly, it will be accepted. And in the majority of situation, luckily, products are non-irritant. And more recently, you had also the 492B, again on reconstructed human cornea. In this situation, you can have a subcategorization between category 1 and 2. So now you have the situation, you have the methods that allow to replace fully the animal. For irritation, there is a lot to do still. A lot has been done, as I show, but a lot to do because there are some specific irritant assays for different regulation. For example, ISO for medical devices. We have some animal assay for oral mucosal irritation, for rectal, penile, for vaginal. But for this tissue, we have some reconstructed 3D models, human reconstructed, and we are working, and different teams in the world are working to try to replace the animal assay. For inhalation also, we are working on this also to replace some acute tox assay based on respiratory exposure. Another very important endpoint, because it concerns a lot of people, the prevalence of the population of allergy is very high. We consider that around 20% of the human population has some allergy towards some different products. So being able to assess this risk is very important. Skin sensitization is based for a long time on animal. We have a different assay based on the guinea pig, the broiler, and the GPMT. In the context of OECD, in the 1999 refinement of this skin sensitization has been done, and the local lymph node assay have been released, which is really highly used now in chemical, not in medical devices. They prefer to use the GPMT especially. So it's based on animal. And I will not develop, but since 2012, with the publication of the adverse outcome pathway, it's a way to describe what is skin sensitization in human. Skin sensitization is not only the redness of the skin, it's other events that lead to appearance of the redness. And often in animal testing, you are assessing only the apical effect. You don't assess what happened before. In predictive toxicology, you are going to preliminary evidence before the adverse effect appear. And thanks to this AOP based on different key events, 3 main test guidelines have been released. Test guideline 442C, D, and E. It's around 12 methods. And thanks to this, skin sensitization can be performed in chemical and in vitro without the need of animal. This is largely admitted. None of these methods are standalone. You need to have some what we testing strategy, and especially to use what we call defined approach to combine the 2 or more free in vitro or in silico methods. Majority of this method in the test guideline are based on in chemical and 2D cell assay. And this assay have some limitation, applicability limitation, especially the 2D cell don't have metabolic activity, so pre and especially proaptants can be detected. There are some limitation for hydrophobic, liposoluble products, and also for unknown composition products. So this limit of applicability limit also the spread and the use of this alternative approach in some regulation. And that's why I think, again, 3D are very interesting, because it can overcome this applicability limitation, and it will open the applicability of in vitro approach to all the chemicals, but also to finished product, cosmetics, medical devices, pesticides, something like this. And this year, a new method has been validated and submitted to OECD. It is the EpiSensa method, which uses reconstructed human epidermis, and it's a toxicogenomic approach based on gene expression. So it's very promising. This method has been developed in Japan with a Japanese model, and I am working now on a Me-Too validation to be able to have a MATEC model in this test guideline. Another point is genotoxicity. Genotoxicity is very important to try to waive some carcinogenicity testing, which are very expensive, time-consuming, and using a lot of animal. And for a long time, some in vitro methods have been developed to assess mutagenicity or DNA alteration or chromosomal alteration. This in vitro are widely used, very important in cosmetic regulation, because you can't do in vivo testing. So you need to have a battery of minimum 3 assay, in vitro 3 assay. In chemical also, you can use it in screening, and if it is positive, you go to in vivo. And for the regulation, you go straight to in vivo, especially for pharmaceutical products. Excuse me. One of the limitations of this in vitro is that you have a high level of false positive because they are based on a cell line, they are based on bacteria, and this is really problematic because, especially in cosmetics, with this high level of false positive, it means that you can't use new ingredients. And in chemical or other regulation, as you have this acid level, you need to go to the animal testing to de-risk your products. And that's why, again, 3D models are very, very interesting. And this last year, 10 years, big validation studies have been performed for 2 assays, especially the COMET assay, which assess the DNA damage, and the micronuclease assay, which assess the perturbation of the chromosome regulation. And this assay, which are now the validation of finished, and it's the peer review is ongoing and should be able to be included in OECD test guidelines soon, I hope. These 2 assays are based on, again, 3D models. For COMET assay, it's based on full thickness, and for micronucleus, is based on an epiderm matrix. So just to conclude, because I see that the time is going on, I have a few minutes. If we have a look of different endpoints, local and systemic, we can say that systemic local endpoint can be now performed without animals. And this, in big part, thanks to 3D models. For systemics, it's more complex. Systemic, we speak more and more about testing strategy, about organ on a chip, about different approaches, toxicogenomics. But in any case, even if we use this, 3D models will be highly important because you reproduce better the human situation. But again, we can be quite positive with what have been performed to replace the animal. Matec in this didn't do all alone, but Matec is a company which tried to mimic all the human organs, especially epithelial tissues, but also more recently the liver, the kidney, and we're working on cardiac tissues, on brain tissues, in order to provide to the laboratory all the tools to develop these methods. Matec is really part of a complex ecosystem of alternative methods with different industry, with different endpoints, safety, efficacy, with different stakeholders and standards, and different countries. Matec is producing in US, historically, in Europe, more recently, is available thanks to some distributor in different countries, including India. But the trouble is that it's often expensive as soon as you have some distributor. And that's why I'm really pleased, and I wanted to be today in India to be able to announce a very, very big breakthrough news about India. It's that we will start next year to produce a local 3D model in India, the Matec models, the same model that are validated for all the methods I showed to you, that will be produced soon in India, thanks to an agreement with Sartorius. Sartorius is an international company that any labs know because they distribute a lot of device and system used in laboratory. And this agreement has been signed recently, and you can see 16th of November. And thanks to this, in Bangalore, next year, we will start the production, and the tentative agenda is based on a training of a Sartorius team who will come in the US to be trained to 3D model production. We'll start in April the on-site production in order to have the first batches released in May, June, and to start to deliver a second part of the year the model to the Indian customers. So it's really, really a huge change. And from what I saw yesterday about some presentation for pesticide for different industry, all is ready in India. You have the CROs, you have the industry, you have the law, the regulation, the standard. And to be able to access this model, I think this can really change the future of alternative in India and accelerate this. Thank you very much for your attention. And I am open to a few questions if it is possible.
Thank you, Christian, for your exquisite, elaborate, and extensive talk on the development of 3D models, with special reference to the reconstructed human epidermis and related models. And I mean, some of the main participants perhaps are listening to a talk of this kind on reconstructed human epidermis in terms of skin irritation and corrosion. They may be listening to for the first time, and they'll be very, very happy to be have been informed like this. And before I throw it open for discussion, I'd like to compliment you for the news that you're going to have this product made in India through your MOU with the Sartorius. And immediately I jumped to telling my colleague, my co-chair, that the product is going to be cost less in India now and be available anytime you want it without any contamination. These are all advantages, big advantages that we will enjoy if the product is made in India, as you would said, with the collaboration with the agreement with the Sartorius. That's a very good company. Like we chosen the right company for us. So having said this, I will throw this presentation open for discussion. And if anyone has a point to clarify, Christian is a very, very renowned expert on this field of reconstructed human epidermis. In several years he's in this field, and he should be able to clarify your points. Yes, you can use this mic.
Hi, Christian. Hello.
Hello.
Hi, I'm Savita. It's truly commendable what you've done, and I particularly enjoyed, you providing a timeline of animal testing history and regulatory history of toxicology. Thank you very much. Okay, so my question was, the development of 3D models now, does that include animal-derived ingredients? And, where are we with replacements and how is that going? I'd love to know more.
There is a lot, again, to do on this. In the case of MedTech, and especially the reconstructed human epidermis, the epiderm model, there are no animal ingredients. It's a chemically defined medium, so we don't have any fetal serum or something like this. After in all the process, I think that for this model, there is no animal ingredients, but maybe some other models could use, I don't know, some coating or something like this, and there is still some job to do. But in the case really of a medium defined, chemically defined medium of culture, I think it's not so important. How can I say? We try to avoid the maximum to limit the use of ingredients. And that's some 3D models. And I know from some other method producer that can use still some bovine serum fetal bovine. And this is really something where they need to change. And for 2D cell culture also, sometimes you need to have these ingredients. And I know that there is a lot of work done on this.
Okay, thank you very much.
Are there other questions? So if there are not any.
Yeah, this is Manny. How are you?
Oh, yeah, Tongla, yeah, I forgot about your online.
Yeah, Christian, a wonderful presentation as usual. You've done a very good job in actually bringing all the regulatory and scientific perspectives together. The one major complaint I have about these skin models is that the metabolic competency, as I know for sure, I've done some work with these skin models, but metabolic competency is not as good as the actual human skin. So that especially puts me into a question that, especially you're talking about immunotoxicity, that many chemicals form reactive metabolites and form DNA addicts or even protein addicts. That actually is the result of the immunotoxicity, which becomes a systemic toxicity. The systemic toxicity cannot be predicted with these kind of models. What are your thoughts about it?
Metabolic activity, I'm not expert in ADME, but from what I know, this is one of the big advantage of 3D models. And this is true not only for skin, but also for intestinal system. And we have a lot of paper about characterization of enzyme of phase 1, phase 2 inside that system, comparison also to human data, and for especially, for example, epiintestinal model, which is a model of intestine. Some pharma company use it to assess this metabolism, something like this. But in any case, it will be always in vitro, maybe not exactly like the human situation. Regarding the systemic, one model will not be able to replace a human body of a full animal. I fully agree. The trouble of systemic toxicology, in my mind, is not only the methods, but it's only to know what is systemic toxicology, what we need to assess, which organ, which parameter we need to assess. So for systemic toxicology, there is a big part of, I can say, basic research, to better know what is systemic toxicology, which organ are concerned, which key event can be very important, and from this knowledge to be able to build some testing strategy where we will combine different individual methods based on the use of some 3D models, because I think 3D can be very interesting. Could be from 3D models of the liver, from cardiac cells, from brain, some different things. And from this to be able to based on a scientific reasoning, to conclude about this systemic and to be able in the future to replace the animal, I think that one model will not be able to use it, but combining different models.
So we have developed a full length skin, full thick skin model like epidermis, mesodermis, and endodermis at our company, Reagin Biosciences. We are not really going for the screening process for the skin irritation and corrosion, as is there are lots of players are doing like yourself, those kind of things. So we are more interested in actually dermatological diseases, applications, and the systemic toxicities of the new therapeutic agents that we want to discover and go after. Okay.
Okay.
If you have any interest, let me know. Yeah.
Okay. Again, I think that the trouble in vitro is the complexity. More, and you know it if you do it, more you have a very complex model that a lot of different type of cells of different compartments, more it's complex to have reproducibility, to be able to produce. So when we are in industrial state and regulatory, it's a balance between very complex system, biological system, and maybe sometimes simpler, but highly reproducible. So it's a balance to find, to be able to have some tools that allow to be predictive of a human. And after you can combine simple model with co-culture, with adding human cells, something like this. But we can continue to discuss. I will be very pleased to discover more about what you are producing in your labs.
Yeah, please. Thank you. Yeah.
So as long as it is a model,
always there is scope for improvement and bringing up new models, and all the best. And I mean, congratulate Christian for the wonderful achievement of matter, and also congratulate Dr. Subramanian Mangala for his Reagene company also coming up with the skin model, the thick skin model, and we wish the company success in the product and its market and its application. We'll be happy to use it in our research. Thank you, Dr. Mangala. Thank you, Dr. Christian, for your wonderful presentation and the sensitization. Thank you so much.
Thank you, sir, for sharing your wisdom and enlightening us on the topic.
I now request Ms. Savita Nutan ma'am to introduce our distinguished guest and the speaker for this session, Professor S. Raisuddin sir, who will be delivering a speech on FISH, an alternative model for study of environmental stress.
Right. Good afternoon, everyone. So Professor Raisuddin is a highly accomplished and respected personality in the fields of toxicology and academic administration. He is the Vice President of the Society for Alternatives to Animal Experimentation, SAAE India. He held prestigious positions such as head of the Center for Translational and Clinical Research, head of the Department of Medical Elementology and Toxicology at the prestigious institution Jamia Hamdard, New Delhi, and research professor at Hanyang University, South Korea. He currently serves as a research advisor and the director of the Internal Quality Assurance Cell at Jamia Hamdard. He has been honored with several prestigious national and international awards. He is editor-in-chief of the Journal of Trace Element Analysis and an associate editor of the African Journal of Agriculture Research.
Thank you. Thank you, Professor Ziaur Rahman and his colleagues for arranging this meeting. My topic will be more academic, and because I am in the university department, so we are really doing research on validation, method development, and we generally guide PhD thesis, undergraduate, postgraduate theses, and whatever comes out of that. And being fascinated with the alternatives, we have a Department of Medical Elementology and Toxicology in Jamia Hamdard, which is a ranked university in New Delhi. And after doing a lot of research on mammalians, slowly being head of the department, I am spearheading kind of some transformation from mammalian models to first non-mammalian model, and maybe in due course of time, no animal model also. Currently, there are three models we are working on with my colleague, Professor Sohail Parvez, and me. He has very good setup for fly lab, that is Drosophila melanogaster, and also C. elegans, which is both are beautiful models. And in my lab, I have previously done, and also currently one scholar is doing PhD on zebrafish model. Earlier, we have a lot of government fundings for doing research on fish as a model for assessment of the toxicity and pollution level of various sites which were there. So you know that there is an environmental stress, and that stress is reflected at various status or strata of the biological system at population level, biological level, physiological level, and it is reflected. And in fact, it is well established fact that when there is a stress is sometimes a kind of the response generating mechanism by which organisms adjust themselves and they become future ready for further stresses and to cope and to mitigate the stress which will come on. So stress is biology or stress is studied in two directions. First is that the adaptive mechanisms to which animals or organisms adapt to the stress, and second is that how to measure different endpoints of the stress. So you know that these stress can last for maybe a long stress and short stress, and many people define for us sometime work stress, office stress, and job stress, family stress, marriage stress. So these are sometimes and at the biological level, sometimes stresses are very for a very short duration. Many of colleagues study the oxidative stress in the biological system or endocrine reticulum stress. They are sometimes created for a very short duration, and then body adjusts through different mechanisms of protective mechanisms or other way to it, and then it is like that. And then stress, once stress is caused, it adjusts and various types of responses are generated, physiological, biochemical, behavioral processes, and many of these stress markers can be studied. For example, if we have a oxidative stress, we can measure it by lipid peroxidation, one of the key indicators, biomarkers of the oxidative stress, or sometime DNA adducts are formed, oxidative DNA adducts can be measured as one of the biochemical responses. Physiological responses also can be measured. We have many types of state, fast breathing, and having different types of behavioral reflects that. And there are four more aspects which are interrelated. One is cellular stress response, and another is homeostasis response. That is, stress has to be adjusted. For example, heat shock protein expression is one of the stress responses. When in order to protect the proteins from heat stress, the heat shock protein 70, 80, and all these types of protein are generated. They act as a chaperone to protect the proteins. And these cell stress responses and cellular homeostasis responses, they work in tandem, and they adjust the biological responses of the organism. And these are various biomarkers which can be studied in the animal models, whatever model, even cellular responses. If you working in vitro, there also it is possible to measure various types of responses. They are measured by release of cortisol, oxytocin, serotonin, serotonin, epinephrine, and brain-derived neurotropic factor, BDNF, saliva, tears, sweat, blood, urine, and immunoassays, and different types of sensors and biosensors are measured or done, and which can be done. And this slide I have taken from some publications, and this stress response, as I mentioned in the previous slide, that there is a this hypothalamus pituitary axis, adrenal axis is there, and it work together, and then stress response is generated, and finally sympathetic nervous systems comes in play. At the measurement level, when we can measure the physiological response, so stress is reflected as a neuroendocrine pathway, where we can measure by cortisol and increased central obesity, increased level of lipids, etc., is reflected. Or it can be measured by various inflammatory responses. We know that by measuring the interleukins, interferons, and other nuclear factors, we can study these, and then finally it is reflected effects on insulin sensitivity and down regulation of insulin stimulating glucose transport and lipid. And autonomic responses of stress can also be measured in humans and animals, or even fish, or what kind of animal models you are using. Some of these parameters can also be studied in vitro. For example, especially the inflammatory responses are reflected in the cell culture condition, in vitro conditions also. So finally, it looks that stress is a very simple response, and we can study, but it is interconnected with so many other processes, biological network in cell health, the body too, and this kind of network and very, very complex responses are generated in through stress. And sometimes we superficially study some specific parameters like oxidative stress parameters like lipid peroxidation and things like that. So fish was my topic title. Then a lot of fish lives in very, very stress situations, especially it is in water, so very less scope to escape from the stress situation. Like if we are terrestrial organisms and we have a scope to escape from the stress situations. If, for example, Delhi was polluted for some time, so in Aligarh we feel stressless or less stress. It's not too much polluted city as compared to Delhi, so we can escape from stress situation. But fish has to live. Either it will die or it has to adjust with various kind of responses. So chemical stresses are there, especially in form of chemical pollutants, which are released by anthropogenic activities and also by some natural processes. Physiological stresses are there, and also perceived stresses like stimulus evoking by some sound, some kinds of other processes. So finally, fish gives some kind of primary response, and then secondary response, and then tertiary response. The primary response is immediate response, which comes then in order to cope the stress. Fish releases hormones, and then levels of hormones is generally increased, specific type of hormone. Then at secondary level of responses, the metabolic changes occur, and at tertiary level, there will be responses in growth and also in reproduction processes. And sometimes it may lead to death, different types of aggressive feeding or no feeding. So as I mentioned that fish lives, fish is also part of biodiversity, our human environment as well. So it cannot escape. So it has to, so therefore fish is a very good model for study of stress, especially the more famous model is zebrafish. A lot of research has been conducted on stress, different types of stresses, their responses in zebrafish. And two types of stresses academically we study. One is oxidative stress, a very well explored area in our laboratory also. We have been working on this field for more than 20 years in many laboratories in my institution and elsewhere. So this is oxidative stress, which everybody knows that creates and affects various processes. I won't much elaborate on that. And then recently, for last 10 years, there is interest in the endoplasmic reticulum stress. And this generates the very, very specific response, that is unfolded protein response, UPR. So this was a recent concept, and scientists have taken up, including in my laboratory, although our initial research is on mammalian models. But one of the graduate students who is sitting here has been working for her PhD thesis in fish model as well, that how these responses can be. So this is showing how endoplasmic response occurs and how different types of parameters or biomarkers which are reflected by UPR response. So there are, although zebrafish is very popular in this part of country, in fact, it is native of this country in the eastern part of a country. It originated from Bengal, Bangladesh area, and then it is spreaded almost whole Europe. America is not so popular, but zebrafish in India, many research laboratories are working at all different possible levels: molecular, cellular, genomics, omics, proteomics, and lots of omics everything is there because genome is available, knowledge is available. But in Eastern Asian countries, there is one more popular model, that is Japanese Medaka, because I was in Korea there, and some time I spent in Japan also. Similar type of attributes or qualities like zebrafish, this Medaka or Orezius latipus, same shape, same dimorphic characteristics, male and female distinguishing, small, easy to sometime reproduce in the laboratory conditions, and the genome is well known, everything. Then in that area itself, there is one more fish, Cryptolabias marmoratus. It is very popular model again in Japan and Korea. And I also worked for it, published many papers when I was in Korea on this species. This is hermaphrodite model. It has male and female characteristics. This third species, very interesting model, lives in the brackish water, that is the mangrove kind of conditions where the high salinity is there, less oxygen is there, and very good stress model. It has a series of applications have been. And then fathead minnow is American model, which American North America especially, and in Brazil and in Argentina also this model, Mexico, they also use this model. So there are, though in our country we generally hear and talk about zebrafish, but besides zebrafish, there are other fish species also which are equally good. But since zebrafish is well settled and well studied and popular model in our country and European and these parts of country, so that is there. So this study was published very recently when they compared the stress responses of three level of organisms. One was humans, and the other was rodent, that is mammalian model, small mammalian model, and zebrafish. So almost similar kind of configurations or organization were there in both the species, and they compared the responses of hormonal responses and in male and female, and a lot of similarities were found in the response. Some differences, except in fish, the male and female response is different as compared in for the cortisol, but the mammalian, the human, and mammalian, that is rodent and fish, some difference. But at the same time, their similarities were also observed in case of isolation, anxiety, and they behave almost similar way when there is a isolation stress or anxiety level, were almost behaving kind of a similar response in humans and rodents and zebrafish. So this knowledge is existing. Zebrafish has been extensively worked for stress research. I won't much elaborate. And since a lot of knowledge is available about neuroendocrine system of zebrafish, so it's a very suitable model for this. So there are various advantages which are described here, and one important aspect is there that the neuron, there are 100K neurons, it's well suited for deciphering entire neural circuit in this, and therefore we can very well study even using plate, 96 well plate or 24 well plates. So these are suitability. Why though I have this my Google profile, so the papers which we published on the fish models are highly cited yet, and though later on some graduate students worked on PhD thesis as well. So these are the top cited papers from our research group. So first one has been cited for 736 times. It is again the fish model study. Second has been cited for 637 times. Again, the fish and stress model. And third paper published in 2000 has 503 citations. So I think our research paper and the area of this research is, though papers were published long back, so therefore the citations are high. So this first paper we published in BBA, that is Biochemical Acta, on the laboratory conditions in fish. Second paper also then published in a taking different chemicals for induction of oxidative state. Then third paper we published, we got a grant from Ministry of Environment and Forest. I think huge grant it was at that time. And then one more grant from Indian Council of Agricultural Research. So they prompted us to undertake research. And here we correlated the pollution level and toxic chemicals which are present in the river water, how the fish model can be used and to predict and to project that what kind of responses are induced. So here we worked on this fish. This fish is very popular, very small, available in market. And we collected this fish from the river waters of Yamuna, which is flowing through this part, going to Mathura and Agra and many Delhi, and then merging at Allahabad with great river Yamuna. So this fish is very prevalently present there. And we sampled this fish from this river water. And we correlated the two types of sites we studied. One is Panipat in Haryana. Another one was Agra in Uttar Pradesh. And both sites are differently polluted. There was high level of industrial activity in Panipat, though Agra is also having industrial activity, but now it is not much because of these restrictions and by the government in order to protect the Taj Mahal and other monuments. So Panipat has many industrial activity, textile and different types of activity. So every parameter we study is popular parameters like SOD, catalase, glutathione, peroxidase, etc., in liver as well as in kidney. And we correlated that the Panipat site always showed a very high level of pollution. Then this lipid peroxidation parameter also we studied in 3 organs of fish, the liver, kidney, and gills. And there also we found this fish as Panipat site is black one and the dotted one is Agra. That all these parameters which indicate the oxidative stress in that level. And then this is glutathione also is popularly studied. It is also showing huge difference between the two sites. So this fish model, not only in laboratory condition, not only for validation studies, not only for regulation aspects, but also can be taken for environmental monitoring. And then there was a fish kill episode that there was a huge fish kill was observed in the Panipat area because of the release of some industrial effluent. And the Environment and Agriculture Ministry of Haryana requested us to undertake the biomonitoring aspects of a study in that area. And there also same kind of, then we, during the fish kill episode, and we compared it after fish kill, that water was diluted and concentration, the pollutants were diluted because of the more flow of water in that area. Then we found that there is a difference between during the episode when we collected the sample and after I think 3 months or so, we again collected water sample and fish samples from that area. And then we found and correlated that if the water pollution was less, then these all parameters were found to be. So we applied this knowledge of laboratory in the field condition. And then we studied the different types of heavy metals which were reported like for chromium and copper, and chromium and this copper again in sediments as well in water. But after the this episode was over, then many of these pollutants that present at very low level, and except the copper was present in sediment again. So again, these various parameters, various biomarkers were correlated during the and after the episode fish kill. So this was study got a lot of interest and publications, citations, and this was there in lipid peroxidation. And we also studied that how the proteins are oxidized. Protein carbonyl is one of the parameters biomarker which is used. So this type of findings I wanted to share, though many of these findings have been published, but here this is a forum where I wanted to say that this fish model is not only useful for the regulatory aspects, also for the method development, but also we can use it comfortably for environmental. My student colleague, Ms. Garima, is working on now endoplasmic reticulum stress and using the zebrafish model, and the compound is the phthalate compound, the DEHP. So I think some research is still under the analysis stages. So nothing much to share on this aspect. So here this thing, and as Dr. Akbar Shah will announce that maybe next meeting will be held in Delhi. So there will be opportunity to visit our place where very well developed, supported by government India projects. This alternative center is evolving. This on the Asophila melanogaster and C. elegans, we have facility, and very soon I think we will also have in zebrafish another alternative model. So again, I must thank for hearing my talk, and if there is anything to, I think this must be the last one. Thank you.
Thank you very much for a very thorough presentation. Does anybody have any questions? I've got a question.
Yeah.
Well, you know, I'm medicine without cruelty, so I have to ask, have you critically appraised the literature for any non-animal methods?
How? Pardon?
Have you critically appraised the literature for any?
I very much appreciate this non-animal model. And one of the colleagues who is sitting here, Shahzad Ahmad, he is working on the in silico model and also working on protein networking, how protein networking can be studied. And he also is working in my lab. So how to, we can correlate. So I think slowly this kind of mission or movement is in place, and we highly appreciate that if there is a non-animal model available, there is a computer-aided system is available, and giving same kind of projections or results. Why don't we are very open to using non-animal, and definitely I appreciate that idea. And in fact, we have a MSc toxicology program, PG course, which have very few universities in India have master's degree in toxicology. And there, with my insistence, we have introduced a 2-credit course on animal welfare in toxicology. And there we also teach one course on how we can non-animal models, even no in vitro, no in vivo, using such kind of in silico or genomics toxicogenomics models.
Yeah, I mean, thank you for that. And it's great to hear, because there needs to be more toxicology programs kind of current with what the future is, looking at
we have revised
non-animal
ourselves, and this course is joined by almost every student. They like to understand it, what is the role of animal welfare in toxicology. Animal welfare means all types of alternatives which are on table.
Yeah. When I did my first degree in genetics, it was one of the first things we learned about is model organisms, and zebrafish was one of the key features. And it was because of the 70% homology genetic similarity that we have. And, later on, and it kind of ties in back with my presentation yesterday, that just because we have this genetic similarity, really what Charles Darwin was trying to emphasize is it's our uniqueness, which makes it our own, our own species unique. So
yeah, every species has its own advantage, and
Yeah.
you have to look into the total picture. Total
Yeah.
picture. Like there was a discussion about the different types of alternatives and all that. Then Professor Vaikunth will vouch. That also we need to know that how much chemical is reaching at the target site.
No, I
Is that
realize.
harmful? So at the final action is at target site. Like if we are talking about mutation, genotoxin, so it has to go to the DNA molecule, cause, adduct, and covalent adduct, covalent binding. So is that much level is going there? Is we need to worry about that? We need to, if sometime, I think that those kinds of consideration we have to do. What is the big picture?
Yeah, no, and I understand. Rigorous testing is definitely required, and I'd like to see more non-animal
Okay.
methods
Thank you.
included. Thank you very much.
Thank you very much. Any questions? Thank you.
Any other questions? No? Okay, we'd like to thank both speakers for the session. It's been very interesting presentation. Thank you.
Thank you, sir, for this wonderful and thought-provoking session. And now I kindly request Professor Akbar Shah, sir, to honor our speaker, Professor S. Raisuddin, sir, with a token of appreciation.
Thank you, sir. I would like to request Professor S. Raisuddin, sir, to honor the chairperson of this session, Ms. Savita Nutan, ma'am, with a token of appreciation.
Now I kindly request Ms. Savita Nudan, ma'am, to honor the chairperson of this session, Professor Akbar Shah, sir, with a token of appreciation. Now I kindly request all the delegates and guests to join us for lunch at the new guest house, and the next session will start after lunch in 45 minutes. Thank you.